बोलो. हॅलो 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 हॅलो. बोल. इधर आओ.
हाँ देखिए, जो यहाँ पे बोलेगा
हाँ
और यहाँ पे बैठे रहेंगे ये इनॉग्रेशन में, इन दोनों को आपको करना है, ठीक है।
कंटिन्यू
जब
लेंगे,
हाँ
जो स्पीच देगा कंटिन्यू लेंगे।
हाँ वो तो कंटिन्यू चलता रहेगा, ये बोलेंगे और ये मिक्सर करेंगे ना, तो ये और ये कवर होना चाहिए। इसके बाद क्या होगा कि यहाँ पे बोलेंगे या यहाँ पे बोलेंगे और वो वाला
फिर कुछ
जो है ना वो cover
या कुछ धागा
ऐसे
मिलेगा
दोनों का मिक्सिंग होना चाहिए।
सेट करना है या टाई वगैरह कुछ। कुछ सूत हाँ, टेबल का ही रस्सियों का तो भी चलेगा। लोगो वगैरह कुछ डालना है।
लोगो एसटीपीसी वाला डाल देते हैं। कौन सा कैंपस वाला डालना है? अपना धनवंतरी आपने किसने यूट्यूब वाला कौन सा डाला है? ट्रस्ट वाला ट्रस्ट वाला
और ये
नितिन भाई टाई से केबल टाई भाई तो भैया ज्योति से आया सर। हाँ निकुंज भाई भैया ज्योति मेरा वाला हाँ हाँ
ઉપરનું ફંક્શન પતે ને પછી આ બધા સોફા નીચે આવવાના છે એટલે તેના માટે અહીંયા જગ્યા રાખી છે તો આ થોડુંક અગાડી જતું રહેશે પછી
પછી બધું કવર નહીં થાય.
એક રો કાઢવી પડશે કંઈ નહીં કરી દેશે સ્ટુડન્ટ કરી દેશે ચાલો નહીં કરે તમારો આ તમારો આ છે ને એક્ઝેક્ટ
હા આજે
क्या फोकस तो कि ढीला मत करना हमें वो डाल दो
मैडम और 5 आइटम मिला है ना मेरा सेशन है तो फर्स्ट से पता चलना तो जाएगा एक मिनट
Hello. Hello.
मिक्सिंग चल रहा है, ये लाइव चल रहा है।
नहीं अभी नहीं चाहिए, कुछ होगा तो मैं
ठीक है, कर दीजिए, बता ही रहे हैं। वेलकम सर, टर्न करेंगे उनको वेलकम करेंगे सर, आ रहे हैं टर्न। क्यों वो नहीं पहले वाला?
पहला वाला वो तो चेंज होगा इसलिए नहीं
आ रहा है क्या अभी बाकी? सर बारीक आ रहे हैं।
फोटो एक करके आ रहे हैं। क्या नाम करना था? अभी लिंक कोई चेंज नहीं हुआ?
नहीं अभी
ये वाला है। हाँ, थोड़ा साइड में
कौन सा?
ये तो लैंप करेंगे।
एक मिनट।
Wishing you all a very delightful morning. Myself, Dr. Shweta Swaminath,
and myself, Dr. Zil Nayak,
are here to host the inaugural ceremony as well as the upcoming sessions of the 7th International Conference on Emerging Trends in Drug Development and Delivery in association with Harrison College of Pharmacy, Urban University, USA, and Gujarat Technological University, Ahmedabad, Gujarat, India. When we talk about the greatest discoveries of the world, we often tend to focus on the modern innovations, looking back to the time when major species of the animal kingdom died of the wound from their respective predators. Humans survived. The reason behind it was that we human beings discovered the magic of remedies to cure, which made us not only survive but evolve as the time progressed. And as we evolved, the drug development also progressed. So as we are here to press the emerging trends in drug development and delivery, I welcome the honorable dignitaries, faculties of STPC family, and delegates to this event. We are about to commence our event by lighting the lamp of knowledge and wisdom. This is a symbolic moment that signifies the dispelling of darkness and igniting of thought and enlightenment. It's my privilege to invite the dignitaries on the stage to do the honors. I invite Ms. Sambhavi Swan to swing the prayer song. All kindly rise for the prayer.
क्या करना ले लो
इधर
या कुंडे तुदुषार हार धवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा
सामने मिहिर
या
सामने
श्वेतपद्मासना या ब्रह्माच्युतशङ्करः प्रभुदिभिः
हो गया थोड़ा सा
देवैः सदा वन्दिता क्षमां पातु सरस्वती भगवती निशेषजाड्यापहा निशेषजाड्यापहा निशेषजाड्यापहा.
Thank you, Sambhavi.
I thank all the dignitaries for gracing the ceremony.
Happiness held is the seed, happiness said is the flower. Flowers leave part of their fragrance in the hand that bestows them. We are glad to share our happiness with our honorable dignitaries by floral felicitation. I invite Dr. N.D. Jivani, President, Sarkar Education Trust, to florally felicitate Dr. Timothy Moore, Professor and Associate Dean for Research, Harrison College of Pharmacy, Auburn University, Alabama, USA. Thank you, sir. I invite Dr. C.D. Salat, former Deputy Commissioner, FDCA, Ahmedabad, Gujarat, and Director of Skill Development Program, SDPC Kim, to kindly come forward for the floral and moment of felicitation of our guest of honor, Dr. Ravikant Sharma, Deputy Drugs Controller, CDSCO India, Ahmedabad Zone. Thank you, sir. I invite Dr. D.L. Sutaria, sir, Secretary, Sarkar Education Trust, to kindly come forward for floral felicitation of our guest speaker, Dr. Robert Rusty Arnold, Professor, Department of Drug Discovery and Development, Director of Research, Harrison College of Pharmacy, Auburn University, USA. Thank you, sir. Now, I invite Dr. M.M. Murvi, Principal, STPC Kim, to florally felicitate our guest speaker, Dr. Jay Chandra Ramapuram, Professor and Graduate Program Officer, Department of Drug Discovery and Development, Harrison College of Pharmacy, Auburn University, USA. Thank you, sir. Now, I invite Dr. Dharmendra Dubey, Principal, SDSET Kim, to florally felicitate our guest speaker, Dr. Elizabeth Lipke, Professor, Department of Chemical Engineering, Auburn University, USA. Thank you, sir. I invite Dr. Darpesh Govil, Chief Facility Management, Ribosome Research Center, Kim, to florally felicitate our guest speaker, Dr. Rajesh Amin, Professor, Department of Drug Discovery and Development, Harrison College of Pharmacy, Auburn University, USA. Thank you, sir. I invite Dr. Anand Deshmukh, Professor, Academic Head and Department Head, Regulatory Affairs, SDPC Kim, to florally felicitate our guest speaker, Dr. Meenakshi Singh, Postdoctoral Fellow, Department of Drug Discovery and Development, Harrison College of Pharmacy, Auburn University, USA. Thank you, sir.
I now invite Dr. N.D. Jivani, President, Sarkar Education Trust, to deliver the presidential address and welcome speech.
Namaste. Today, Chief Guest Dr. Timothy Moore, Dr. Ramapuram, Dr. Robert Rusty, Dr. Elizabeth, Dr. Amin, Dr. Manisha Singh from USA, Dr. Sharma sir, Drug Controller, Deputy Drug Controller, Ahmedabad Zone, Dr. Shelat sir, retired Deputy Commissioner, Food and Drug Control Administration, Gujarat, Dr. Sutariya, Secretary, Sahkar Education Trust, Dr. Nulvi, Principal, Dhanvantari Pharmacy College, Dr. Darpesh Gohel, TFM RRC, Ribosome Research Center, Principal and faculties from various colleges and STBC, and my dear delegates. I heartily welcome you all in 7th International Conference on Emerging Trends in Drug Development and Delivery. The purpose of organizing this conference is to share knowledge and insights on the latest advancement in pharmaceutical science on the theme of new drugs, biomaterials, and delivery system for better human and animal health. During these 2 days, you will learn latest advancement in pharmaceutical technology. I would like to extend my gratitude to our keynote speakers, panelists, and presenters for sharing their expertise with us. I would also like to thank our organizing committee, sponsors, and partners for their tireless efforts in making this conference a success. Thank you all.
Thank you, sir. I now invite our beloved Principal, Dr. M.N. Mulvi, to introduce our honorable guest to the audience.
Good morning. Let me welcome on behalf of Sri Sahkar Education Trust and Dhanvantari Pharmacy College all the dignitaries on the dais, the delegates, and the sponsors who have tirelessly worked for this event to happen. So it is an immense pleasure to introduce the dignitaries and the distinguished speakers who are going to share their experience and their knowledge with the delegates. So the key speaker, Dr. Tim Morey. To read his biography, he had joined Harrison College of Pharmacy in 2016 in Drug Discovery and Development Department, and 2019 he has been awarded with W. Walker Professor and Associate Dean for Research Program. Before joining Harrison College of Pharmacy, he has served in National Institute of Health in various positions like Medical Officer, Chief of Lung Disease and Branch for Lung Division. Also, he was Executive Director of NHLBI, Science Moving Towards Research, Translation and Therapy Program. So, you will be hearing when we will have their speech. I will be just giving you a 2-line introduction of the speakers, so I will not be taking much time to read the biography in detail. And they are going to have their plenary session. That time we will be knowing each speaker's biography. Now the second speaker, Dr. Jai Babu Ramapuram. He is the one who could help us in initiating this program, and it was his idea that I will get my colleagues to your campus. Let us have some program. So with this, let me ask all the delegates to give a round of applause for Dr. Jai for making this event happen, and we are here to celebrate this event. So now Dr. Jai Chandra Babu is currently working as Professor Graduate Program. He is a PG and PhD from BHU, Banaras Hindu University. Then he moved to postdoc at USA. Then later he joined Auburn University as a teaching faculty. Now he is working at the capacity of professor in drug discovery and development. The next key speaker is Dr. Robert Arnold Roski. So he did his PhD from University of Buffalo in 2004. and he had postdoctoral training at Cancer Therapeutic and Non-Invasive Imaging at UB Roosevelt Park. Then he has joined initially with the industry as an R&D head, then later he is serving at Harrison College of Pharmacy at the various, right now he is serving as a Director of Research and Development and Support. The next key speaker is Elizabeth Lipke. She is a chemical engineer. Elizabeth Lipke, she is awarded with the Dorothy Stafford Utla Endowed Professor at Auburn University. Now she is serving as a professor in Chemical Engineering Department. Her specialization is 3D development, human engineering, cardiac tissue, and she will be working on a lot of medical devices kind of research. So definitely I welcome you, madam. The next distinguished speaker, Dr. Rajesh Amin. He did his PhD in 2003 from University of Detroit, Michigan. Then he completed his postdoc training in metabolic therapeutics at Pfizer. Then he joined Harrison College of Pharmacy as adjunct faculty, then as adjunct faculty at School of Kinesiology. Apart from these things, NIH has funded him $6 million for his research activities as a grant. So with this, I welcome Dr. Rajesh Amin. Now, Dr. Meenakshi Singh, she is a postdoc under Dr. Rajesh Amin. She is working as a postdoctoral fellow. She did her post-graduation from NIPER and also the PhD from NIPER. Then she has joined Auburn University as a postdoc. With this brief introduction, I thank all the delegates, all the speakers, and all the faculties and the coordinators and the team who has tirelessly worked for this event to happen. Thank you, Vanina.
Thank you, sir. I now invite Dr. Darpesh Gohel to deliver the presentation of Ribosome Research Center to the audience.
Good morning all. So first of all, I would like to thank all the sirs and guests sitting on the dais, as well as all the organizers, professors and students to make this conference happen today. Let's quickly introduce Ribosome Research Center services and capabilities. So let's, who is going to click the slides? Please, thank you so much. Yeah, so Ribosome Research Center is a global contract research organization which is mainly providing services to different industries like pharma, medical device, cosmetics, Ayush, specialty chemicals. Actually, the whole facility is spread over 1 lakh square feet of the area. These all services are being provided through different departments, as mentioned here, like chemistry department, who is looking after all the physical studies, chemical characterization studies, method development, validations and all. Second is microbiology department, which is having biosafety level 2 laboratories, where we are providing all the microbiology related batch clearance testing as well as different environmental sample, water analysis and other testing services. Next is in vitro pharmacology and toxicology department. Actually, this department is the start of any preclinical testing of any pharma products or medical device or agrochemicals or anything, because this department is taking care of all the cytotoxicity as well as genotox studies like chromosomal aberration test, bacterial reverse mutation test, micronucleus test. These tests decide the future of molecules. If any molecule is genotoxic or creating any mutation in the DNA, then that molecule will not further progress. Right, so this department is very important, where we have more than 12 cell lines in-house library. Next is in vivo pharmacology and toxicology department, where we having barium maintained small animal facility with 55 clean rooms. Each and every room is equipped with 0.3 micron HEPA filter plus 100% fresh air. So this is the largest facility across the Gujarat, or you can say in India, where we are having more than 50-55 clean rooms in a single sort of facility. Next is test item control office. This is the department where we are receiving all the test items, or you can say drugs or medical devices or any other chemicals to be tested. Here we are providing unique test item code as per the RRC list. Then we are storing that particular material at different storage conditions like room temperature 2 to 8 degree, -20, -80 degrees centigrade based on the type of product. And then archive department. This is very important department because after completion of all the GLP studies, we need to archive all the dry material as well as wet material. As a part of dry archive, we are maintaining all the paper documents. And as a part of wet archive, we are maintaining all the tissue, slides, blocks, specimens, as well as representative sample of test item for next 9 years. So anytime during this next 9 years, any auditor or any sponsor can come and audit the study. Then next is our information technology department, which is mainly looking after all the data backup through 3 different servers. First is our local data backup system, which is through Lenovo server, supported by 2 NAS systems. One NAS system is out of the facility, so we can ensure utmost security of all the GLP or even non-GLP data. And this data backup is taken at every day. Every 15 seconds or we are taking backup of all the data. Apart from this technical departments, we are also having certain quality management system which is mainly governed by our ISO 17025:2017 for medical devices, microbiology and chemistry, and OECD GLP also, because this is both are international quality systems which are being required for submission of any of the toxicology or safety studies. These are the list of certifications or accreditations we have. It covers FDCA, CCSCA, that is mainly for animal experimentation, Schedule L1 GLP we have Ministry of IOS license, ISO 17025:2017 and CDSU. So CDSU is having MD40 certification, that is mainly for medical device testing for India. Now at Ribosome Research Center we have more than 500 to 600 instruments. Here I have captured major like list of instruments. In chemistry, like currently we have more than 80 instruments. So major instruments from there are LCM, SMS, GCHS, FTI, UV, HPLC, UPLC, DSC, flash chromatography, NMR. So using these instruments we can provide different kind of testing which covers bioanalytical services, it covers all nitrosamine or genotoxic impurity quantification and chemical characterization of medical devices and many more. As a part of pharmacology and toxicology department, I have listed a few important instruments here, but in this department we have more than 250 instruments. So like NMS system with video tracking, grip strength measurement system, motor activity measurement system, digital plethysmometer, rotor, this kind of instruments basically required to go for neurotoxicity estimation, neurobehavioral estimation, functional observational battery measurement system, mainly for repeat dose study as well as reproduction study to be submitted to the regulators for getting approvals of the product. In our in vitro department, we have CO2 incubators, multimode plate readers, RT-PCR, PCR, electrophoresis, gel-based system, which are mainly required for all in vitro testing as well as certain gene expression studies also. I will not take much of your time. I will quickly running these slides just to save the time. As a part of in vitro and in vivo efficacy studies, these are the list of models we have already worked with. Apart from that, as you know that for any efficacy model, there are no certain specific guidelines. So we can customize each and every efficacy model or efficacy studies, either it's an animal model or in vitro model, and we can offer to the clients. More importantly, we are also working for efficacy testing of all cosmetics. This is very interesting studies actually. They all are in vitro studies. using different cell lines for mainly for anti-hyperpigmentation, anti-stretch mark, right? You can say for fairness creams and all. So these kind of testing are required to prove that actually that particular product is working or not, and that is also through certain scientific pathways. So all these kind of testing we are providing there. As a part of in vitro safety or toxicity studies, we are doing all genotoxicity studies, certain in vitro alternative testing like DPRA, skin irritation using reconstituted human epidermis, TER, PK, in vitro release test. All this kind of testing we are providing as a part of in vitro studies. This is the list of studies which we are providing as a part of our in vitro services. In vivo services. It includes single dose PK studies to 2 years of combined chronic CAR-C studies. So we cover all the animal services as a part of in vivo. One of the major portfolio of Ribosome Research Center is medical device testing as per ISO 10993 guideline. This is also a regulatory required specific testing. So we cover all the biocompatibility testing, which includes in vitro study as well as animal studies. And these all are mandatory studies as per ISO 10993 guideline, because without this you cannot register your medical devices to regulators. Apart from this, we are also providing services for chemical characterization testing, including biological evaluation plan and reports. Maybe these things are new to you, but these are the important necessary part of regulatory services or regulatory requirements. And mainly for students, you can have your bright future with this industry also, because this medical device industry is not that much famous, right? Actually, this industry also requires engineers who are having computer engineering or chemical engineering or mechanical engineering. So that kind of designations required in medical device field also. And most important is risk assessment plan and report. So that is also part of one of the most important services we are providing. As a part of chemistry department, we are providing all the dose analysis testing, bioanalysis, all pharmacopoeial based testing we are providing, 5H analysis, DSC, XRD, NMR. We are also doing certain method development, validation as well as tech transfer to certain companies. As I said, as a part of microbiology department, we are providing all the services as per Indian Pharmacopoeia, British Pharmacopoeia, USP as well as EP. Major services are like BET, MLT, sterility, environmental sampling, water analysis, and recently, if you know, there is one species called as Burkholderia cepacia, which is mandated by US FDA to be evaluated as a part of certain gel or oral mouthwash and all. So that also we are providing. So this is about a physical facility of Ribosome Research Center. This is the main building. This is a dual chiller system. You will not find any kind of laboratory where you will have a dual chiller system. Most of the time you will have one single chiller system supported by DG or USB. Here we have dual chiller system supported by DG as well as USB. So you can understand how much important, because the HVAC is most important in any laboratory. And to save HVAC, to save animals, to maintain specific temperature, humidity and pressure in the laboratory, this supporting system is very, very important. This is the snaps of our animal facility. In animal facility, we have a dual corridor facility. So like it's a clean corridor, followed by animal holding room, followed by return corridor. And the air flows is from the positive to negative pressures. So every room is equipped by such system so that we can control all the backflow of air to the animal facility. We also have a individually ventilated caging system in our facility. So this kind of cage is mainly required for immunocompromised mice. You must have heard about the nude mice, kid mice, right? Athymic nude mice. So that kind of animals are being used, and we can perform any kind of studies, including carcinogenicity study using transgenic animals in this kind of facility. This is the supporting cage changing station. So wherever we have to use this system, this cage changing station is being used to maintain the aseptic condition for each and every cage, because this each and every cage is having different environment. They are having a HEPA filter on each and every cage, so we can maintain separate environment for each and every cage here. These are certain important instruments. This is, I said, animal system, which is followed by video tracking system, and all the movement of animal is being covered in that software. So you can have all the movement, behavior of animals through video, and then you can analyze that using the software. This kind of instrument are mainly used for the learning and memory of the animals. So this is a histopathology lab as well as clinical pathology lab. These are the most important part of any animal testing, because see, of course, if animal testing is completely closed, then we are the happiest person, right? But as of now, it is not possible, because lot of regulatory, including India, requires animal testing. Because as on today, we are not that much developed that we can completely demolish the animal testing. So that's why as of now, regulatory also asks for different animal studies, right? So this is the set of instruments for histopathology. This is the set of instruments for clinical pathology, where we can perform all hematology, clinical chemistry, urine analysis, coagulation meter, histopathology examination. These are certain snaps from the important instruments from the chemistry. As I said, we have LC-MS, which is also supported by APCI group for nitrosamine impurity quantification. This is GCHS, where you can perform chemical characterization, or we can say ETO residual analysis. Most of the medical devices are sterilized using ETO, ethylene oxide, right? So we have to perform residual analysis for ETO also. So that kind of testing we can perform using this GC. These are like multimode plate reader, RT-PCR, where we can perform certain gene expression studies as well as in vitro analysis. This is our microbiology lab. In archive, we have this kind of compactors. So because we have to store all the documents for next 9 years, so we can store in this kind of compactors. So that's all from my side. Thank you so much. You can contact me through email or phone call if you need any further information. Thank you.
Thank you, sir.
We have with us Dr. Vinit C. Jain, Principal, Bhagwan Mahavir College of Pharmacy, to grace the events. I request Dr. Anand Deshmukh sir to florally felicitate Dr. Vinit C. Jain. Thank you, sir. We have Dr. Nidhi Chauhan, Principal, Lakshmi Narayan Dev College of Pharmacy, Belgaum, Bharuch. I request Dr. Uttam More sir to florally felicitate ma'am. Thank you, sir.
Now we have a message from Dr. Rajul K. Gajjar, Vice Chancellor, Gujarat Technological University. Let's listen.
Good morning. Namaskar. Respected Shri Prafulmi Pancheriya sir, Dr. Montu Patel, all the distinguished speakers from Auburn University, all the team members of Dhanvantari Pharmacy College, and dear participants. It is an honor to be able to address a gathering of such distinguished participants as well as speakers on a topic that stands at the frontier of modern science. As we strive to improve health outcomes for humans and animals, innovations in pharmaceuticals, biomaterials, and delivery systems will be reshaping the way we tackle disease and enhance the well-being of all on this earth. Advances in genomics and bioinformatics have paved the way for therapies tailored to individual genetic profiles. A prime example is the gene therapies that will likely address the disorders like sickle cell anemia. Another groundbreaking area of mRNA-based vaccines, which COVID-19 has shown us, can now be extended to combat diseases such as malaria and cancer, so prevalent in our part of the world. Biomaterials are revolutionizing medicine by offering innovative solutions for drug delivery and tissue engineering. They will hopefully reduce the frequency of doses and improve the patient compliance, which is so very important. The use of nanoparticles in oncology have tremendously helped minimize the side effects on healthy cells, and therefore nanomedicine improving the drug efficiency by 40% compared to the traditional methods. Looking to this scenario, the smart drug delivery systems such as wearables, which are now quite popular, and all the innovations that keep on happening day to day, especially in the pharmaceutical world. We need to come together. We need to look at all the advancements, but they are not without challenges. High costs, regulatory hurdles, and ethical considerations must also be addressed. But the potential to alleviate suffering and improve quality of life makes this pursuit worthwhile. Let us all come together, look to the future, collaborate between researchers, industry leaders, policy makers, and together we can translate these emerging trends into impactful solutions. I congratulate the entire team for such a wonderful conclave, such a wonderful gathering, whereby we will get a way forward, and we hope that you will come up with the best of the outcomes and follows. Thank you. Jai Hind, Jai Jai Karbi Gujarat.
We also had a message from Dr. Montukumar Patel, President, Pharmacy Council of India, wishing us all the best for this event, but unfortunately he was not in the moment to make it. Okay.
Now, I invite guest speaker, Dr. Timothy Moore, Professor and Associate Dean for Research, Harrison College of Pharmacy, Auburn University, Alabama, USA, to address the gathering. Sir.
Thank you very much. I was getting my presentation loaded up, took the opportunity to do that at this moment. So, but at this moment, I believe I am supposed to just address the crowd for this wonderful attendance. By the way, the room is full, and that is so exciting to see. I think those of us that have traveled from Auburn University to be here, after we've been talking about this for quite some time in our offices and our telephone calls and our emails to arrange this, certainly when we walk in this morning and see so many people here that are so eager to learn, to be educated, to pursue degrees in pharmacy, to pursue degrees in engineering, and then importantly, hopefully at the end of this conference that we've put together, to pursue how that collaborative work between pharmacy and engineering will lead to impactful research. And that's the core of why we're here, and that's the core of why our faculty from Auburn have taken the time, because they're dedicated to that mission. We're dedicated to drug discovery and development. We're dedicated to all of the things that need to go into the research I was really impressed by listening to the ribosome research presentation a minute ago, and all the detailed elements. Just to re-emphasize those, particularly to students in the in the group that are here today to learn and to understand. Drug discovery and development takes time. It takes precision. Right? It takes care. But most importantly, it takes passion and enthusiasm. Right? We can't forget those elements when we talk about going into this type of career. Because our goal, our mission, I know at least speaking on behalf of our folks and from Auburn University, is that drug discovery and development, combinations of pharmaceutical engineering, and all those disciplines that are part of that academic environment, what they're doing, we're doing that so that we can improve the health and well-being of the people around us, right? And so hopefully we'll keep that in mind as you're going to hear a lot of really great science from the presentations, and you're going to hear a lot of great speakers, and hopefully, I know we're going to see a lot of great presentations and posters and other things tomorrow, I believe, right? So I'm very much looking forward to engaging with everybody in that aspect. So thank you very much for having us here.
Thank you, sir, for your words of wisdom. It will be a great pleasure for us to listen to you again in our future events. Now, I invite our guest speaker, Dr. Jayachandra Ramapuram, Professor and Graduate Program Officer, Department of Drug Discovery and Development, Harrison College of Pharmacy, Auburn University, USA, to address the gathering.
Namaste, good morning everyone. I'm glad to be here again within few months to see you all, many of you that I have met you before. And I'm very much impressed during last visit when I was here and got the tour from your college administration to your college facilities as well as ribosome research facilities. And so I'm highly motivated and enthusiastic to come back and hope that we'll participate with our research collaborations in the future. So with that motivation, I requested some of my colleagues to come and visit and witness what Dhanvantari College of Pharmacy would offer in terms of research collaborations and research facilities. So I'm very thankful to my colleagues who have agreed to participate and join, willing to travel all the way from US to here and find the free time and release time to make the trip here, and we are here, we are very glad to be here as a part of the, as a group here to participate in these scientific events. So without taking much time, we would be in the coming next few hours or so, we'll be having some exciting scientific talks from our group, and we'll be also enthusiastic to look at the research that has been going on at your end in terms of your posters and presentations. And I wish you all the best for this event. So far, I've been witnessing it's going extremely well, and I wish you a good event. Thank you.
Thank you, sir, for your influential message. We are eagerly looking forward for your plenary lecture. Now, I invite our guest of honor, Dr. Ravikant Sharma, Deputy Drugs Controller, CDSCO India, Ahmedabad Zone, to address the gathering.
Delegates on the dais, speakers from overseas university, students, ladies and gentlemen, very, very good morning to all of you. Good morning. Good morning. Yes, came to? Good. I am Dr. Ravikant Sharma. I am a Deputy Drugs Controller and I am representing Central Drugs Standard Control Organization, that is the National Regulatory Authority of India. My earlier speaker has mentioned the regulatory practices. You are students, you are the young future of our country. Many of you can join the regulation, many of you will be the entrepreneur, many of you will choose and opt this academic profession. How many of you know about regulation? How this drug is regulated, under which act? So, let me apprise you about the regulatory things. I will give you an overview about that. National Regulatory Authority, that is headed by Drugs Controller General of India, that is the Central Licensing Authority. We have 7 zonal offices, we have 8 sub-zonal offices across the country, and we have the 7 laboratories for testing of drugs. As you know that we have federal system in our country. The regulation, some duties are dealt by the central licensing authorities and few are dealt by the state licensing authorities. Central licensing authority is responsible for giving the permission for clinical studies, for BAB studies, to enact the law, to make the amendments, and the state licensing authorities mainly govern the sale and distribution of the drugs. Friends, our healthcare system is supported by 2 verticals. One is the pharmaceutical and the other one is the medtech sector. If you see the pharmaceutical sector, that is well established. We have $50 billion economy as of now, and we are anticipating it to grow up to $150 billion less than decade of time. We are producing the generic medicines on the affordable rates. We are supplying to more than 200 countries. We are producing more than 60,000 preparations of 60 different categories of the generic medicines. We are at number 3 by virtue of the volume, and by virtue of value we are at number 14. So friends, this is the commitment of our industry. Every 5th person in the world is consuming our medicine. So give a big round of applause to our industry. And out of this export, if I apprise you about the Gujarat state, there are more than 3,000 units, pharmaceutical units, and more than 25% export of total export is exported from Gujarat. So this is a very, very good achievement from the Gujarat state. Now Gujarat is leading in case of API, formulations, medical devices. We are producing the heart valve in our country, the TAVI, and that is from Gujarat, Shahjahanand company. They have discovered that TAVI cost, heart valve cost is 20 lakhs rupees in our country if it is imported. But if you manufacture in our country, the cost is 5 lakhs rupees. We are manufacturing the intraocular lenses, we are manufacturing the orthopedic implants, we are manufacturing hypodermic syringes and needles, so many things in the medical devices. Right? So this is the contribution of the Gujarat state. If you take the medtech sector, the total economy is around $15 billion, but we are expecting to grow up to $25 billion within 5 years of time. Now, if you see in the medtech sector, 80% of the medical devices as of now is imported from the developed countries. So we have not a caliber in such field. So I wish that our youth generation should come in the medtech sector. Because if you see the disease burden in India, that is characterized by the known communicable diseases like diabetes, like cancer, like cardiac diseases, and India is the diabetic capital of the world. So we need to grow the advanced technology for faster diagnosis, for correct diagnosis, because we know that without diagnosis, treatment is blind. After correct diagnosis, only therapy can be started. Friends, if you want to grow, the pharmaceutical industry or the medical device industry, we have to keep in our mind the 5D formula of our Prime Minister Shri Narendra Modi ji. And what is the 5D formula? The number one is the discovery, the innovation, the research, the pharmaceutical development. By the virtue of generic medicines, we cannot achieve our goal of 150 billion US dollar. Can we achieve? No. We have to develop the new therapy, the cell therapies, genomics, recombinants, biosimilars. Then only we can achieve. And today, there is a conducive environment. We have 7 NIPERs in our country which are funded by the Department of Pharmaceuticals, Ministry of Health and Family Welfare, 700 crores to them for the Center of Excellence. So this is the time. Now the college, academic institution, pharmaceutical industry, they come and they should have the collaboration to produce the good product as per the market needs. If I recollect the COVID situation, we must have gone through very tough time. During this COVID time, our industry came forward and they discovered the new products like tocilizumab, like favipiravir, like ivermectin, like remdesivir. This is the requirement of our country and our industrialists, our scientists, and even COVID vaccine. We discovered indigenously. Likewise, in the medtech sector, we researched out about the ventilators, the oxygen concentrators, and we supplied not only to our country, we also cater to the other countries also. Because we believe that there's a one earth, one family, and one future. So first D is the discovery, the research and development. The second D is the deregulation. Deregulation means there should be a harmonization of the regulation. There should be no over-regulation. The regulation should be clear, understandable, articulated, so that our industry can grow up. And right now, we have the new drug clinical trial rules which demonstrates the requirements to conduct the clinical trial studies, the preclinical studies, phase 1, phase 2, phase 3 and phase 4, PKPD studies. It gives the requirements about what is the sponsor responsibility, what is the ethics committee responsibility, what is the exclusion criteria, inclusion criteria, and to evaluate the dossiers. Whenever a manufacturer is submitting the clinical trial dossier, it is evaluated by the 50 subject expert committee of the different fields. So whenever they are submitting the protocol, it is evaluated. When there's what is the inclusion, exclusion criteria, what is the design, whether it is randomized, double blind, and the report is also evaluated by the subject expert committee before giving the approval in the market. So we have the medical device rule in place because all the medical devices are under regulation. If you see from tongue depressor to class A, class B, class C and class D device, these are categorized on the basis of the classification and the high risk. So all these medical devices are under regulation, and class C and class D medical devices are regulated by the Central Licensing Authority, and class A and class B licensing governed by the State Licensing Authority. Now the third is the democracy. When we enact the law should be in consultation with the association, with the manufacturer, as per the perspective of Indian conditions. That's why our Prime Minister says, Sabka Saath, Sabka Vikas and Sabka Prayas. When we make the law, making of the law is not an easy task, it's a very difficult task. We have to take the the comments from our academia, hospitals, our this other regulatory authorities, associations, and the rules are enacted. Fourth D is the demography. Means whatever the drugs we are manufacturing, it should be made available to the poorest of the poor. The availability, affordability, and accessibility of the drugs. Right? This is the best example of the COVID vaccine. We, supplied 1.2 billion doses of the COVID vaccine free of cost to poorest of the poor, and that is not an easy task. So give a big round of applause to the government also. And the last but not the least is the demand. if you have the good quality, then your product will be accepted everywhere, not in India, but every part of the world. Quality is not an act, it is a habit. And quality always is determined by three factors. One is the quality of raw materials. The second is obsolete of the old equipments and machinery. And third is the training. Training to our employees who are responsible for distribute, packing and sale of the products. Right? So these are the five, this 4D formula, what our Prime Minister Modi ji is narrating for growth of the industry. In the last but not the least, I will say with the one quote of the Churchill, the highest, what is the greatest assets of the country? The healthy people. If we have the healthy people, that is the greatest assets of the country. Thank you very much for your patience. Thank you, and I wish you all the very best for the grand success of this event, 7th International event. Thank you so much.
Thank you so much, sir, for your inspiring words. We are grateful to have you today.
No duty is more urgent than that of returning thanks. So I invite Dr. Anand Deshmukh, Professor, Academic Head and Department Head, Clinical Regulatory Affairs, STPC, KIM, to propose the word of thanks.
Yeah, good morning to all the dignitaries, esteemed speakers, guests, faculty members and students. It is indeed my privilege and honor to propose a vote of thanks on this remarkable occasion of 7th International Conference on Emerging Trends in Drug Development and Delivery, organized by Sri Dhanvantri Pharmacy College, in collaboration with Harrison College of Pharmacy, Auburn University, USA, and Gujarat Technological University, Ahmedabad, Gujarat, India. First and foremost, I would like to extend our deepest gratitude to our chief guest, Dr. Ravikant Sharma, Deputy Drug Controller India, CDCO, Ahmedabad Zone. Sir, for your valuable insight and guidance on the drug regulatory process. And of course, to Dr. Narendra Modi's 5T plan, and in detail and inspiring each one of us for continuous research and taking the nation first. We are deeply grateful to Dr. Rajula K. Gachar, Vice Chancellor of Gujarat Technological University, for her recorded message, which have added immense value and encouragement for this gathering. I would also like to extend my thanks to Dr. Montu Patel, President, Pharmacy Council of India, for sending his best wishes. I would like to extend my heartfelt gratitude to our distinguished speakers, Dr. Timothy Mori, Dr. Jayachandra Rampuram, Dr. Robert Rusty Arnold, Dr. Elizabeth Lipke, Dr. Rajesh Amin, Dr. Meenakshi Singh, for sharing their presence, which promised the enriched scientific session. I express my heartfelt gratitude to Dr. N.D. Jivani, President of Sri Sarkar Education Trust, Dr. D.L. Sitaria, Secretary, Sarkar Education Trust, Dr. M.N. Nolvi, Principal of Sri Dhanvantri Pharmacy College, Dr. Shidi Selath, former Deputy Commissioner, FDCA, Ahmedabad, and Director of Skill Development Program, Sri Dhanvantri Pharmacy College, Dr. Darpesh Gohel, TFM, Ribosome Research Center, along with Dr. Dharmendra Dubey, Principal, Sri Dhanvantri College of Engineering and Technology, and Dr. Rose, the Principal of International School, Dhanvantri Campus, for their guidance, leadership and support in making this event a reality. A sincere thank to our collaborator, Harrison College of Pharmacy, Auburn University, and Gujarat Technological University for their invaluable partnership in fostering this platform for international collaboration. I also extend my appreciation to the organizing committee, faculty and volunteers whose hard work and dedication have ensured the seamless execution of this event. Finally, I thank all the participants and attendees for their active engagement and enthusiasm to make this conference a vibrant, impactful and a success. Once again, I thank everyone for being a part of this significant event. As we embark on this journey of exploration and collaboration, I am confident that the knowledge shared here will inspire innovation and progress in drug development and delivery. Wishing you all a fruitful and enriching experience. Thank you.
Thank you, sir. Now it's time for the tea break. The session will commence in 15 minutes, so I request all of that right back.
Your phone.
दो हो गया ना चाय पानी कर दिया
बंद करोगे चालू
चालू रखना
આપણા કમિટી બોયઝને બોલાવો બધાને કે સોફા નીચે મૂકવાનો
अच्छा इसीलिए सबको ऐसे जैसे
हमको किसी ने पता नहीं कि ये ऐसे लाइट की तो ये मैं रोज ऑलरेडी फिर कैसे कर पाऊंगा अभी वो मैंने बाईपास कर दिया है अभी वापस
देखो यार चालू
ओए અહીંયા આવ ને આનો
આ બાજુ મૂકી દે ને
બીજામાં જો એ ખરાબ हाँ चालू 15 मिनट में बोलो आयरो 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 બંકર ને એરું
अब आ जाओ
સ્ટુડન્ટ બોલો સ્ટેજ કમિટી સ્ટુડન્ટ મોકલો તમારા હા તો એમને મોકલો હા હા એમાં કાયરા ડિસિપ્લિન વાળા જોઈએ હવે આ બધું કાઢલા પણ નામ બી કાઢલા
एक्स्ट्रा जो तो लाइब्रेरी में जो एक्स्ट्रा હોય ને
મારા ખ્યાલથી ક્રિસ એડજસ્ટ કરતા
एक्स्ट्रा
टेबल से ऊपर जो टेबल आ ले लेना
ની એ ઓકે ની આ આગળ રાખો પછી આવી રીતના બેસવાનું છે
ના ના હવે આવી રીતે નથી બેસવાનું હવે અહીંયા કોઈ જ્યાં બેસવું હોય ત્યાં બેસે
ઓકે ઓકે બસ તો
સાચું જ કે
આમાં તો મૂકી દો ને બે બે બનાવી દો नहीं भेज भेज आपसे सर भेज भेज आपसे भेज बेन सेटिंग
કઈ કઈ કરીએ અરેન્જમેન્ટ થઈ જશે મારા વચ્ચેની વચ્ચે તો ચેરપર્સન જશે કઈ નહીં ચાલશે ચાલો ને પાંચ આ સોફા રવા દે એક બાર જવા દઈએ પાંચ સોફા અરેન્જ કરીએ
એક ત્યાં આવી જશે ને એમાં ત્યાં આવી જશે
એટલું આગળ ભાઈ છે હોવું જોઈએ ને નહીં મળે દિલીપભાઈને પૂછ્યું દિલીપભાઈ કોઈ ક્લોથ છે હું ટેબલ પર લગાવવા
રેડ કલરનું મોકલાયેલું ને
કાં છે
અહીંયાથી જો નાલી મેડમ પૂછવું પડશે એટલે ક્યાં મૂકવાનું છે બધા એની ઉપર લગાવવાનું
हेलो हेलो हेलो
एक रेड भेजा था वो
सर वो नहीं अच्छा लगा
दूसरा अभी वहाँ से भेज देते हैं
हाँ
આ તો બહુ ખરાબ લાગે ઉત્તમ સર કે આ જ કોમ્ફર્ટેબલ આ તો જો આખી અહીંયા વચ્ચે 4% હતું
नहीं
है ચાલી આવો
नहीं तो सिंगल टेबल भी आपने
मैं तो कहूँ टीपॉय मुझे तो चाले टीपॉय पर कैसे ना लखाई के नहीं लाओ जो अरे का मैं चेयर पर बैठे नहीं हूँ
अरे वहाँ पे है वही
टीपॉय लाओ नहीं बोला 30
ऊपर से नहीं लगे
क्या
नहीं वो छोटा वाला
इधर करेंगे
नहीं वो टीपॉय पे लिख पाएंगे कि नहीं वो चेक करना
कुछ करने वाला ऐसा
हाँ अभी नहीं परफॉर्मेंस नहीं 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 अभी वो होगा स्पीकर आएगा ये ना लेक्चर ये नहीं पहले मोटू लावे के बंदा सेम हाइट है लेना पीछे
हाइट से दोनों
लावी जाए
अरे
ये इनको पीछे एडजस्ट करवा भैया आप थोड़ा पीछे लीजिए ना वो इनको एडजस्ट करना पड़ेगा
ये पीछे
बैठने वाला नहीं है
उठा के वहाँ पे रख दो
नहीं नहीं इधर ही रख दो उसके ऊपर हम सब सामान रख देते
लाओ तो फिर कर ला
हैं 4% इधर कहीं अबे इधर सब पूरा ये कवर है 4% देखेगा कहाँ से सुनेगा कहाँ से हाँ
तेरे टेम्पोररी ये मुका दो किसी यही
मैं क्या बोल रहा हूँ इसको थोड़ा पीछे लीजिए वो कैमरा पीछे ले रहा है
नहीं वो तो ले रहा है लेकिन 4% को सेंटर में बिठाना होता है जो अभी लेक्चर चालू होगा ना वो उसको वो सब नहीं नहीं तो पहले देखा है नहीं कैमरा में 4% डाल दिखाएगा नहीं नहीं तो अभी देखा से कैसे बीजे अरेंज करूँ पड़ता सोफा तड़क पछाटी लेने ये कैसे अरेंज करती है आमाज मूकी दे ना सोफा थोड़ा पछाटी ले ले
उस साइड ही कर सकते हैं
हाँ इधर अरेंज कर देते हैं हाँ यहाँ का थोड़ा पछाटी ले अबे कहीं नहीं तो स्टार्टिंग में
वर्षम यहाँ से और दीवार में लगा सकते हैं देखा मान लीजिए
हेलो हेलो माइक टेस्टिंग हेलो
बस ये मुके दो टेबल ये मुके जाओ ये तेरे बंदे उसको ये में जो है
हेलो
हां थोड़ा क्रॉस करिए
हेलो हेलो
चेयर आवा तो ये है वो रोटेट था और ये दिलीप भाई ने पूछा चल चल दिलीप भाई देखा तो नीचे ओ भाई पहले सुना मैडम
हेलो 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 ओके
बोलो पंकज भाई
हेलो
अरे यार
हेलो
हेलो
हेलो हेलो हेलो हेलो आवाज आ हेलो 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 माइक टेस्ट testing.
हाँ बोल गुरु
Hello, 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 hello. 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 Hello, hello, mic check. Hello, hello, mic check, hello.
Thank you very much. Again, it is my honor to be here today, to be a part of this symposium, this excellent conference. I was thrilled earlier this morning before we took our break at all of the great words that were said regarding the enthusiasm around this conference and symposium. And now I think what we want to do, obviously, is move into the portion of the conference and symposium and hopefully be able to exchange ideas. And that's the purpose for these events, is to be able to exchange ideas, to be able to... What? Right here? Better? Okay. Thank you. Just point me in the right direction and I'll get there eventually. There we go. But hopefully everybody heard what I said when I was standing over there. Okay, great. But anyway, I look forward to what we're going to be able to discuss, like I said, for the exchange of ideas that we're going to have, and certainly when we have that opportunity to do that tomorrow with poster presentations, and really be able to get one-on-one time with everybody who's doing this exciting work in drug discovery, who's chosen to go into those fields. What I'm going to do with my time now is just set the stage a little bit, but I am going to talk mostly about Auburn University. My purpose is to try and orient you to who we are at Auburn University, guide you in the course of how we're doing things at the university that's related to drug discovery and development. And then I will take a little bit of time just to make sure you're reoriented with our leaders in drug discovery and development who will be providing their lectures following me. But Auburn University, we are located in Auburn, Alabama, United States. Okay? If you were to try to get an idea of what that looks like, this is what it looks like. This, of course, is a pictorial of the southeastern portion of the United States. If I were to put my pointer right about here, yes, right about there, that would be Washington, D.C. That's where Washington, D.C. is located, the capital of the United States. And so you go through 1, 2, 3, 4, 5 states southwest from Washington, D.C., the capital of the U.S., to get to our state, where we're located, which is the state of Alabama. The state of Alabama is right in the center of a group of states that's commonly referred to as the southeastern United States region of the United States. And then if you look a little bit closer, the state of Alabama, right here, just on the eastern border of Alabama, right in between Georgia and Mississippi, you'll find our university, Auburn University. We're about 100 or so miles south of Atlanta, Georgia, which is the capital of Georgia, and just a little bit east and north of the capital of Alabama, which is Montgomery. And that's just a picture of the front door, or the front of our access to our beautiful campus there. So when we talk about Auburn University in the future, because we expect collaborations to come out of these things, so hopefully you'll continue to hear Auburn University, you now have a picture in your mind of where it's located. Our university in and of itself is composed of 13 different colleges, and these are listed here. Agriculture, architecture, business, education, engineering, wildlife, environment. Our graduate school is an independent college that helps oversee all of the Auburn University graduate programs and the independent colleges. Human sciences, liberal arts, nursing, our College of Pharmacy, science and math, and our veterinary medicine school. Between all of these colleges, we award or we offer more than 150 undergraduate majors. All right? housed in 12 colleges. The graduate school, which is a college that doesn't award independent degrees, but the other 12 colleges award disciplinary degrees. And then our graduate school offers a large number of degree programs as well as accelerated bachelor's and master's programs. We also are proud of our international education and our engagement in international education. We have many abroad programs that are based in Auburn to help allow students, faculty, and others to exchange education and ideas and gain experiences across the world, and vice versa. We also do very well at helping international students that join us at our university learn English if they don't know English, or they learn it better, I guess I would say. So we have a good, strong program in that. Many other international programs at our university, including culture. We believe culture is very important and foundational when we talk about international engagement. And then, of course, we just recently started a very formal Korea Center with some formal interactions and relations with some of the universities in South Korea. Two things here to point out are new university bachelor's degrees. And those degrees, those 150 degrees, majors that I mentioned a few minutes ago, these are the two newest that were just approved and have been put into place, and students have been joining these degrees beginning just this fall, so just in August of this year. Okay? And the reason I want to point these out is because these types of degrees and what we're doing here are very appropriate to the types of things we're talking about at this conference, which is integration of health ideas, interdisciplinary work, drug discovery and development, and the idea of One Health. Okay? Now they're found in two separate undergraduate degrees that we just began offering. The Public and One Health degree, and you can see its description, oops, sorry, hit the wrong button. Described here, it is actually emanating from our College of Veterinary Medicine. Right? But it partners with many other faculty across colleges in order to talk about public health. But the philosophy underlying that degree is the One Health message, which is it's human health and well-being, but it's also animal health and well-being, right, across the spectrum. And then the other newest degree at Auburn University is the degree that we have produced at the Harrison College of Pharmacy, which is the Drug and Biopharmaceutical Sciences degree. This is the first undergraduate degree, truly an undergraduate degree, BS, leading to a BS degree, from the Harrison College of Pharmacy. It basically is designed to create the next generation of pharmacotherapy specialists and pharmaceutical scientists. It's a pipeline to our other degree programs in our college, including the PharmD, which is the clinical degree in pharmacy, as well as our graduate programs, masters and PhDs in our pharmaceutical sciences. And we believe this has a tight link that's going to be developed over the course of time with industries, particularly pharmaceutical industry that will be seeking these types of students that have this type of degree and expertise. So what about some other facts about Auburn? By the numbers. Auburn student profile. Our total head count enrollment, as of most recent numbers, 34,145 students at Auburn University. They come from all 50 states in the United States and 108 different countries in our student population. Ratio, about 51% to 48% female to male in the student body population. Of the total 34,000, 27,907 are there for undergraduate degrees. New freshmen coming in every fall. Right now, averages about 6,000 students that start at Auburn University every fall. And Auburn University is one of the more competitive colleges that you can apply to, and particularly not only just in the Southeast, but in the country. Is somebody here from the University of Alabama just shut that down? My folks with me. I don't know what to do. Anyway, I was going to continue to, I don't hate for time to get away from us, but was going to continue to talk a little bit about Auburn. Here we go. I have to wait on the computer probably. Anyway, what I was going to say was we're a competitive institution, and so we're very proud to say that the best and the brightest students and future graduates join us at Auburn. Okay, this is where we wanted to continue looking at our faculty profile. Faculty, at Auburn University, we have about 1,400 or so total faculty that are teaching in all those colleges that I mentioned. 89% of our faculty have the highest terminal degree they could obtain. So we have very highly educated, well-trained faculty in addition to our competitive students at Auburn University. And it works out across the board, across all the colleges, to be about a 1 to 20 faculty to student ratio. That's an undergraduate headcount number. Importantly, Auburn University is designated a Carnegie R1 Research Institute, which means Auburn is an R1 institution and we have very high research activity. This is the top classification that you can get for research institutes in the country regarding research activity. So we have a lot to live up to with that designation, meaning that we have excellence that we strive for and who we're bringing into our college, both in terms of our students and our faculty, and we have high expectations that we're going to produce the best research and most impactful possible. And I'm going to hopefully lead you to show how we do that, and I'll go ahead and give you a hint. It's by collaboration. That's one way. And just to kind of also put some perspective around Auburn in relation to other things, we are the #1 university in our state, in Alabama, and we're not the only university. There are other universities, but we are #1. And relative to our research rankings, at last ranking, we're in the top 11% of all research institutions in the United States. Continuing along some of those numbers, we rely on U.S. News and World Report a lot to help determine where we are in our country nationally. And in U.S. News World Report, we are number 93 in all of the universities located in the U.S. 51 top public school in our country. Ranked number 29 in terms of first-year experiences. So those students that I told you that compete to try to get into our college and our colleges and universities, they like being there in their first year, so that's a good thing. And then we were also given a designation of most innovative school, number 63 in the nation. We also rely on another ranking system called the Princeton Review, which kind of dives into things like the student life and student experience at Auburn University. And other aspects of what our college, what our university portrays. For example, in the country, according to Princeton Review, we have the #2 best athletic facilities. We pride ourselves in academics, but in the southeastern United States, we pride ourselves in athletics. College football season in the U.S. is coming to an end right now, and it's very exciting. It's not as exciting for Auburn this year. Our team didn't do as well. But we do currently have the #2 basketball team in the entire country. So we're doing well in that. Health services, again, these are rankings that we're proud of regarding the quality of students and student engagement in life that we try to offer at Auburn University. But the one that really stood out in the Princeton Review of Rankings is the one that's right here. And because it stands out, I made my own slide about that. The happiest students in the nation. Can you believe that? That's actually, that's a ranking. But how to reflect that data, if you were to ask anybody from any place they were at, they're going to say, I want to be from Auburn. Because apparently we have the happiest students in the nation, in the U.S. And I know I'm laughing about that, but that's something to be proud of, because that's the type of culture, that's the type of environment that we want to foster. That's the type of collaborative environment that we want to engage in when we partner with others in very specific activities and programs. Recently, Auburn University just completed a 10-year strategic plan. We were fortunate to start a new administration with a new president of our university just 2 years ago, who, by the way, is an engineer, and who, by the way, is a chemical engineer, and is now the president of our university. Going through strategic planning takes a lot of work, takes a lot of guidance, takes a lot of sight and vision, and it takes a lot of aspiration. And so we think our strategic plan is very aspirational. And the reason I wanted to bring that up is because it's not that someone made the plan at the university and then told all of us this is what we've got to do. We're the ones that made the plan by working together across those colleges to say, this is where we want to go, right? And that's the way it should be done ideally. And in doing that, what emerged were things like, I showed you 2 undergraduate degrees, okay, that now exist at Auburn, that didn't exist before, but the purpose and nature behind those degrees is understanding cross-disciplinary work, cross-disciplinary team concepts, right? So one of the things that's really emerging and really hopefully concepts we'll get in this symposium of the next couple of days is this idea of teamwork in drug discovery and development is absolutely necessary. Okay? But our strategic plan basically has 5 major goals. We're not going to go over all these goals, but just to highlight the high-level places that Auburn University and our colleges are wanting to go. We want to make sure we continue to maintain an exceptional student experience. Right? We want our students to always be the happiest, right, every year. But right up there at the top, we want to make sure we're engaged in impactful research and creative scholarship. Right? So that Auburn University continues to move up other rankings, the world rankings. We have high aspirations to try and move in that direction and become certainly more visible in things we do. And research will be a key driving force for us to be able to do that. Commitment to excellence and innovation, catalytic engagement, and then something we call distinctively Auburn. There are things about our history and tradition that make us distinctively Auburn. And we want to make sure that's always something that's we're engaging in and being attractive to. All right, so moving from kind of the university level to the college level, again, 3 of the 4 faculty members that will be presenting today are from the Harrison College of Pharmacy, including one of our postdocs that will also be presenting in that group, and our other esteemed leader that's in the group is from our College of Engineering, and I'll mention that in just a second. But I will start with the College of Pharmacy and just a little bit of information about the Harrison College of Pharmacy at Auburn University. We're basically driven by 3 overarching principles: research, education, and outreach. Outreach can also have other names. Outreach can also take the form of clinical practice, service. That's what outreach is. One thing I didn't say a few minutes ago is that Auburn University in the United States is what's called a land-grant university, meaning that it is our mission as a land-grant university to always find ways to be given back to the community and providing service to those for which we are educating. All right? And as a component of that certainly falls under what we do in clinical service and what we do to try to provide healthcare, health and well-being, not only through the College of Pharmacy, but through our College of Veterinary Medicine, colleges of nursing, and in collaboration with each other. But the Harrison College of Pharmacy at Auburn has been training pharmacists in pharmaceutical sciences for more than 135 years. So we've been around a little while. We've had an opportunity to make a lot of mistakes in how we educate and correct those and get better, right? And same thing with research, same thing with how we deliver healthcare. All right? So having a nice track record means we learn from things and we always try to improve and iterate and make things better in the future. We offer these degree programs at the Harrison College of Pharmacy. First and foremost, I guess, our staple degree is the Doctor of Pharmacy, which is the PharmD degree. This is the healthcare professional degree, and we pride ourselves in educating what we call the practice-ready graduate, such that the pharmacists in the United States, when they graduate with a PharmD degree, pass their licensing exams, they're capable of working in collaboration with taking care of patients in our healthcare system. Right? We offer the bachelor's degree program now that I mentioned a minute ago, which is tailored towards developing new generations and people who are working in pharmaceutical sciences, specifically drug discovery and development. Our graduate programs, we offer master's degrees and PhD degrees. These are obviously our research-based degrees. We offer some tracks in that. We have a health outcomes research policy program for MS and PhD students to pursue. And then we have our collective drug discovery and development graduate degrees, which break down into these disciplines of medicinal chemistry, pharmaceutics, and pharmacology. And then finally, we have a number of dual degree programs, where some students who are pursuing a PharmD degree may also pursue a PhD degree. Some students who are pursuing a PharmD degree might pursue a Master's of Business Administration or a Public Health Master's, right? So we try to encourage that for those students that want to obtain that dual education pathway. So it really makes them robust when they graduate from our college in terms of thinking what they might want to do professionally. 3 academic departments: drug discovery and development, health outcomes research and policy, and pharmacy practice. Some numbers about the college specifically. Currently, as of this start of this semester, we have 490 total students enrolled in our PharmD program, the professional degree, leading to professional degree pharmacy. Those classes sizes are broken down here on the left. The ratio is 3 to 1, easily, female to male, and who are pursuing our PharmD degree in our College of Pharmacy. And then the in-state versus out-of-state ratio is also shown here. So we have a lot of people who are from our state of Alabama who pursue healthcare degrees at our College of Pharmacy, but we also have a number of students that come from elsewhere, and that includes international, some international students, a few international students. The other feature of the Harrison College of Pharmacy is that we have the main campus for education that's located at Auburn University in Auburn, Alabama, but the College of Pharmacy is a statewide pharmacy program. And what is shown here is the state of Alabama, and each of those stars indicate where we have faculty that are working in healthcare environments or in communities doing pharmacy practice, and our students in their 4th year will rotate in these places and locations across the state. So we're a statewide College of Pharmacy. All right, so we have our presence, it's at Auburn, main education there, and a main campus also located down here in Mobile, Alabama. But we have pharmacists that are working and students that are learning all across the state all the time. Our graduate and undergraduate enrollment, our graduate program total is about 45 students. 10 of those are pursuing that health outcomes research and policy degree, and then the other of those 35 or so, are in our drug discovery and development master's and PhD programs. However, we currently only have 1 master's student, I think, in drug discovery and development. Everybody else is in a PhD track right now. And then just to show the undergraduate program, remember I mentioned it just started this fall, and we started off with 9 students to enroll in that program for a new degree at a big university like Auburn. 9 very brave students, because it was a new degree being offered at Auburn University. This time, though, as we're looking ahead to next year, we already have had over 50 applications for next year to come into that program. Because I think what will happen over time is the value of pursuing that type of science in drug discovery and development is going to become more and more attractive. The Auburn Harrison College of Pharmacy is a member and fully accredited by the Council for Pharmacy Education in the United States. Just to put it in perspective, every state and Puerto Rico as a territory in the United States has one or more colleges of pharmacy located in those states, with the exception of Alaska, Delaware, or New Hampshire. They're the only ones in the U.S. that don't have a college of pharmacy. What we've seen in this decade is that there have been a tremendous number and growth of colleges, schools and colleges of pharmacy across the country. So that just at the turn of the century, schools and colleges of pharmacy numbered about 80. And currently, ACP accredited colleges and schools of pharmacy are 139. So there was a growth, particularly in the early 2000s. But one of the things also about that growth is that there's a little bit of imbalance, and we actually have seen over the past few years the numbers of students entering into pharmacy school have actually gone down a little bit. So it's always a balancing act to find out the number of students, supply, demand, market forces, and other things are very important in driving that. But the reason I showed this is that at least you get some perspective or idea when I show you this next slide, which is the rankings. Again, we rely very heavily on U.S. News and World Report rankings. We have other sources and metrics for rankings, who we are and how we relate. But we are currently, as of this year, the number 29 College of Pharmacy in the United States. So that places us easily in the top 20% of all pharmacy programs in the United States. We think that's not bad. We think we could do better. You always think you can do better rankings. But that's actually pretty good because of all those, that southeastern region I showed you, composed of all those other states. There's about 41 colleges of pharmacy, because we include Puerto Rico in that. We're number 6. So regionally speaking and nationwide, Auburn University is doing quite well and recognized for what we're doing in terms of college pharmacy education. And one of the things that we want to continue doing well is this, which is impactful research and scholarship. We have a number of activities, a number of research programs, a number of faculty who are engaged in research, and we have a number of opportunities for students to also pursue research ideas. But we also want to make sure that we particularly are focused on a mentality when we're in our college and our research and when we do our collaborations, is that we'll always have the end in mind, which is improving health. Okay, so that's a key thing for us. That's what kind of differentiates us from like a college of science and math, where it's biology or fundamental things. That's important. I'm not saying it's not, but oftentimes that's who we want to collaborate with, because we can work together in ideas that are going to lead to thinking about how to improve health and well-being. All right. So that's kind of the mantra, is that our impactful research and scholarship, we always want it to be translating pharmacy research into healthcare. Over the past several years, we certainly have seen a growth in our research in our college and what's been coming out of our college in terms of research. This is just one metric, and that's the number of publications. These are index publication ranges going back from 2013 to 2023, looking at a 10-year increase and a 10-year upswing in the amount of scholarly activity that's coming out of the Harrison College of Pharmacy. And there are a lot of areas that this covers, but if you were to break this down some, one area that we certainly could point to where we've been making tremendous amounts of gains and our visibility with what we're doing in our research is in drug discovery and development. Also, one of the metrics that we kind of look at is how well are we doing at bringing in funding to help support our research efforts. This graph on the right shows basically our extramural funding, and in particular, how much funding we're bringing in as a college, an independent college, but looking at the National Institutes of Health as a target, because that's what we want to be looking at, because that funds our health research. As you can see, we've had a steady increase over 10 years in our extramural funding. And as we've increased our funding, we've also been able to increase how much we spend in our research efforts at the College of Pharmacy. And what helps drive that is that our faculty, who I showed you, who've been producing a lot more publications in what they're doing, are also submitting a lot more proposals for grant funding. In fact, that number has doubled as well over the past few years. This is the real reason, I guess, I wanted to show this slide, is because, as I mentioned, drug discovery and development efforts have been leading a lot of our key indicators that have been going in the right direction for the Harrison College of Pharmacy. And this is a freestanding research building for our drug discovery and development department. This is about a 35,000 square foot building, 3 floors, containing all the modern laboratory facilities and equipment needed to help support our faculty here conducting drug discovery and development research. We also have another building where we have drug discovery and development research being conducted. It's a little older, but between those two, we have tremendous amounts of activity in drug discovery and development. The backdrop, though, that also, again, helps us frame what we're trying to do when we talk about drug discovery and development is a translational research mindset. Translational research, translational science is very specific when you start talking about it in those terms. It means the thought processes from discovery all the way to application. Okay? And when we talk about health, and we talk about health care and our research that helps improve health care, a translational mindset is very important for us and how we begin to frame how we want to advance our research mission. Translational spectrum, and you heard earlier one of our invited guests talk about regulatory sciences. Translational sciences and translational research often is going to encompass having to have an understanding of regulatory sciences, because it is ultimately how you start thinking about taking an idea that turns into a molecule and a drug that makes it into the market that actually is used to help treat somebody's disease or prevent a disease, right? And these are just the stages of translational science, translational thinking, where you have very basic discovery science that are kind of fall under the clinical animal studies that a lot of us do. And then you have to take it a step further to translate all of this discovery into humans, where you then begin to start thinking about engagement and interaction with regulatory sciences and moving something from a bench to a bedside, right? And that's T1 translation. T2 translation is where you begin thinking about not just the effects it may have on humans, but certain patient populations that you're trying to develop a new drug for to treat them therapeutically. T3 then is after you've had all of these clinical trials that you heard mentioned earlier today conducted so that you finally have a drug that maybe has come from an idea at the bench to make it into a marketplace to be able to treat disease. Okay? Then it doesn't stop there. You have to continue to evaluate how effective that drug is, maybe against a newer drug that comes out. So that's this later stage of translation cycle, which is translation into practice. Understanding that maybe there's a new drug coming along, you test that against an older drug in the market, right? And then finally, this what we call this translation to community. And that's just a continued evaluation of any drug that has been put into the marketplace that has been in use, and monitoring it to make sure it continues to have safety, right? We want drugs, first and foremost, way back here to be safe, right? To be used safely. And then we can make sure they're used effectively, right? And so all along the spectrum, safety is something that's always considered when you talk about translational research. The drug discovery and development component of that is what is driving that early stage translation, all right, as shown here. All right, and this again just kind of identifies some of the key components of drug discovery, which now you start hearing a little more disciplinary terms that you're familiar with, like medicinal chemistry, omics, pharmacology, right? These disciplines that are going into an understanding of these first phases of drug discovery and development. What I'm showing here basically is just, you know, the different components that lead to preclinical testing in the spectrum of drug discovery and development pathway, and then into ultimate clinical trials. What's populated here are the names of faculty that are working hard in this area at the Harrison College of Pharmacy. Currently, they're in drug discovery and development work, and kind of the area that you would maybe characterize them as being working in that space mostly. But what you can see is that our academic department, from an academic department standpoint of drug discovery and development, we have a nice scatter of people who are working all along this early preclinical development drug stage. And the other thing about this, it's not specifically listed here, is that they all have very different disciplinary expertise, such that the ideal spectrum of moving from this end to that end with this expertise and it being multidisciplinary is that they can work in teams, right? In theory and in reality, they should be able to think about team concepts and work together to accomplish developing a new drug. This just breaks it down in another format, but what's added here that's different from that previous graph that I showed is kind of some topical areas for which our faculty, students, and others at the Harrison College of Pharmacy are working on in drug discovery and development, in case anybody here has similar interests, which you probably do. Cancer, diabetes, cardiovascular and infectious disease, drugs of abuse, substance abuse. We have very strong and emerging programs in those areas. Neuroscience is a big area, and you're going to hear some of that later this morning, or this afternoon. We have a little bit of work in pulmonary, not that much, honestly. And then inflammation and ocular diseases. So a number of disease states that our faculty are engaged in terms of seeking new understandings of the mechanism of disease, but coupled to the thought process of drug discovery and development. And the important thing also is that while there's applicability to different diseases that we think about and things that we want to try and take care of from a healthcare standpoint, many of the underlying principles for drug discovery and development are agnostic, meaning there are still things you have to do no matter what. So there are principles and processes that we try to educate and learn along the way, whether it's cardiovascular disease or whether you're developing something in cancer. Okay? Relatively speaking, this again just kind of shows a bigger picture of the Auburn University landscape and the amount of engagement that we have with the National Institutes of Health, which is the major funder in the United States for biomedical and pharmaceutical research. And what you'll find is that basically we have five strongly clustered colleges at Auburn University that all compete nicely and receive National Institutes of Health funding for their research. The College of Pharmacy, of course, being one of them. If I were to have shown you this graph or something like this a few years ago, those bars would not be that high in those areas. What we've seen has been a growth in our ability to be funded by the National Institutes of Health for the type of work we're doing. And a lot of that has been driven by our faculty who are working in drug discovery and development. But if you look at other colleges, and I'm going to call out the college that's right here, College of Engineering, they've also been very successful in achieving funding for their work that's related to health. Okay? And we're going to hear some of that, lessons learned about that in later lecture. But this naturally lends itself when you see these types of data, and everybody's working in the same goal or mission to try and obtain National Institutes of Health funding for biomedical research, that opportunities for collaboration and cross-disciplinary work here are very strong, right? And in fact, in reality, the reason that you'd see those bar graphs that have lifted up, including our own, is that there has been collaboration that's already been going on. And that's why the numbers are going up, right? And that's actually what's driving it. In fact, those examples of that are given here. We've had success in drug discovery and development, in pharmacology and omics-based discovery and development. And when you break it down as to how that success is achieved, there have been collaborating units. College of Engineering, Vet Med, even liberal arts, College of Science and Math, and College of Pharmacy. Same thing can be said about tissue engineering and 3D cancer models. College of Pharmacy, College of Engineering, College of Veterinary Medicine, College of Human Sciences. Same thing can be said about natural products and botanicals research, which we have a program in that which is well funded by the National Institutes of Health. Collaborations, very important across there. Drug delivery, imaging, and therapeutics. The way that we've been successful in doing impactful research, collaborating. College of Engineering, Vet Med, College of Science and Math. And that includes also the other aspects of drug delivery in a more advanced fashion, such as 3D printing and advanced manufacturing work. College of Engineering, College of Veterinary Medicine, College of Pharmacy. So if you'll notice that in almost all of those success examples of collaborative efforts that have led to research impacts for us and our drug discovery and development experience, translating to successful outcomes such as increasing our funding to continue that type of work, you might notice that the College of Pharmacy and the College of Engineering seem to be partnering in many areas there. And the College of Engineering at Auburn University, in its own right, is extremely successful college, and I didn't want to overlook that fact because they are certainly one of those colleges that is elevating Auburn University's stature very high in the rankings. The Samuel Ginn College of Engineering, in terms of their reportability, 53rd among all engineering colleges, which was up from the previous time that was taken in that ranking in 2023. And as you know, I mentioned 139 colleges of pharmacy across the United States. In terms of engineering, there's a lot more than that. A lot more. I don't know the number. Dr. Lipke may do that. A lot. So that's pretty impressive. So right there, in terms of the College of Pharmacy, our best neighbor, even on the campus, if you knew the location of wherever they were located, doing tremendously good things. They're also ranked 30th among the public universities in the United States. Okay. They are a full-fledged, all-encompassing College of Engineering. And these are the numbers of students that come to Auburn University to go to engineering school there every year. If you hadn't already forgotten, I showed you 34,000 or so total numbers of students that started this year, all right? Or that were enrolled in Auburn University this year. When you add up all of these students that were present in engineering degrees, which are outlined here, all these different degrees in engineering that are offered, and you include the numbers of students that were in graduate programs at the same time, okay, that totaled to be 6,331. So 1/5 of all of the students that are coming to Auburn University are in Auburn University and stay there they have some contact or touch point with engineering pursuits. Okay? And then lastly, regarding this message, what is it that engineering is wanting to do when they talk about their scholarly advancement and their research activities? It's this. These are their areas that they have strategic research focus in. Advanced manufacturing materials, cybersecurity and intelligent systems, infrastructure, energy and environment, and one that has been emerging, and certainly Dr. Lipke has been leading in that effort, is biomedical and health systems engineering. So, that's okay. I only had two slides left. And that was going to be this next one. Let's just kind of sum up. I showed you a lot of data. I talked a lot about Auburn, but I want you to know about Auburn, and I want you to know about where we're at, what we're doing. I want you to know that we have very much shared values and interests from the colleges of pharmacy at Auburn, colleges of engineering at Auburn, and what your colleges are doing here in pharmacy, and obviously recently in engineering. We all have motivation for drug discovery and development research. And one thing that we have found in our experience that has been helping us be more successful in drug discovery and research is collaborative multidisciplinary partnerships. Collaborative multidisciplinary partnerships, right? And this conference was founded on a recognition of collaborative multidisciplinary partnerships and the importance for the next phases and emerging trends in drug discovery and development. And in particularly, we're going to talk, or you're going to hear from us on our side about some of those areas that are going on in the pharmaceutical sciences and in engineering, and particularly biomedical engineering. Okay? So our goal now, obviously, to listen to our next speakers, but I encourage you to think. And when we engage in poster sessions tomorrow, and we have the opportunity to come around and be excited by what we're going to see on posters, we're going to be thinking about collaboration, multidisciplinary science, and partnerships. Okay? So, what we want to do, the goal is put here. We want to reinforce translational mindset thinking that's needed to support drug discovery and development research. Recognizing multidisciplinary approach is critical. We want to identify ways that advance drug discovery and development in an academic setting. Most importantly, we want everybody in the next couple of days to try and engage in the dialogue around this. I know these faculty members were introduced earlier, but this is a transition slide, and to let us all know that we're about to be able to hear from them directly, and their talks are going to be better than mine, I promise you that, because it's going to be more exciting about the work they're doing. And as you can see, they're going to cover a wide variety of different areas to be able to do it. In addition, Dr. Singh, who works with Dr. Amin and postdoctoral fellow in that, will be delivering a talk as well. So you'll get to hear from five of us. And with that, I don't really expect any questions, but if you do have questions, then hopefully you can catch me, particularly if you want to talk about drug discovery academic setting or something related to Auburn University or in that regard. And I believe now, I thank you very much. And I'll turn this over to, I don't know, who gets the microphone next?
Thank you, Tim. Really, it was great to hear about the Auburn University, what really great work it is doing in pharmacy, engineering, and other disciplines like agriculture, human education, animal science. Definitely, it was a great job Auburn is doing for United States. And I also wish to have a long-term collaboration with our organization where we may think of a dual degree program where our university has already given a green signal, where 3 years the graduates can study here and 2 years in Abon, and we can have B Farm and M Farm integrated degree in 5 years. That kind of course can be designed, and we can think for a future and long-term collaboration. With this, really I thank your valuable information about Auburn University. Thank you very much.
Thank you for letting me share.
Thank you very much, sir, for sharing your profound knowledge with us. I kindly request Dr. Timothy Moore to mark his esteemed presence by signing the board. Thank you, sir. I request Dr. M.N. Mulvi, Principal, STPC King, to felicitate Dr. Timothy Moore with a memento as a token of gratitude and love. Thank you so much, sir. Now, we will be moving towards the next session, which is a multi-omics approach to treat metastatic drug-resistant prostate cancer by Dr. Robert Rusty Arnold, Professor, Department of Drug Discovery and Development, Director of Research, Harrison College of Pharmacy, Auburn University, USA. We have Dr. Nidhi Chauhan, Principal, Lakshmi Narayan Dev College of Pharmacy, Bolao, Bharuch, to chair the session. Let's welcome Dr. Robert Rusty Arnold to deliver the lecture. Sir.
A very good afternoon, everyone. So it's a privilege to introduce our second session's guest speaker, Dr. Robert Arnold. Dr. Arnold earned his PhD from the University at Buffalo in 2004, followed by postdoctorate training in cancer therapeutics and non-invasive imaging at UB and Roswell Park Comprehensive Cancer Institute. Before attaining academia, he worked at Wyatt AS Research in liquid and parenteral R&D. He began his academic career as an assistant professor at the University of Georgia. In 2012, he joined Auburn University's Harrison College of Pharmacy, where he is working as a full-time professor and served as a director of research development and support. He held adjunct position in pathobiology at AU and small animal medicine and surgery at the University of Georgia. He is a senior scientist at UAB Comprehensive Cancer Center. His research program focused on developing and utilizing nanomedicines and alternate dosing schedules to enhance cancer treatment. He is particularly interested in engineering composite multifunctional nanomedicine that exploit tumor differences within tumor microenvironment to optimize drug exposure profile. His work involved creating targeted therapy and nanomedicines to treat primary and metastatic cancers and improving in vitro methods to mimic cancer biology. He served as a PI, MPI, co-PI and collaborator and awards totally over $22.8 million, communicated 5 book chapters and 90 publications and over 250 meetings, proceedings and abstracts. Currently, he serves as a chairman of AU Faculty Research Committee and part of the leadership team for nanotechnology, pharmacodynamic, pharmacokinetic and drug metabolism, and communities within the American Association of Pharmaceutical Science. He is a reviewer in journal NIH and DoD and editor in several journals. So I warmly welcome sir for the today's session on a topic precision-based drug delivery, a multi-omic approach to treat metastatic drug resistance prostate cancer.
Thank you very much for that kind introduction. I'd like to thank the organizing committee and thank you very much for that kind introduction. I'd like to thank the organizing committee and the leadership for inviting us to come to India to give this talk and to meet with both faculty, your leadership, and students and attendees who are here. Today, I'm going to give you a brief introduction of some of my research. I'm classically trained in drug delivery, but my education was largely in doing pharmacokinetic analysis, modeling, and simulation, and I've tried to do something a little bit different. I will tell you I'm not an expert in formulations. Rather, what we try to do is alter formulations to improve exposure-response relationships. And what I've done in the last 10 to 15 years is started to incorporate more molecular approaches to do this. So a lot of what you're seeing was not done by me, but done by my graduate students, done by postdocs, and actually done by some very highly skilled undergraduate researchers within my lab. So as Dr. Moore had given, again, the US, we are located here. For those who don't know, Auburn is a slightly confusing area. Our mascot is the tiger. So we have Albie the tiger. He consistently ranks as the top mascot in the country. But when we see people, we scream War Eagle. So just to give you an instance, while walking through the New Delhi airport, someone came up to me and said War Eagle. So that's how we greet each other if we've got our logos on. And it is interesting because wherever we go in the world, we do run into people who are either affiliated with Auburn or actually came from Auburn as well. And I won't go into the story, but if you ever come to Auburn, we can educate you into why we actually scream War Eagle, even though our tiger is actually our mascot. So what I'd like to do is further this idea of drug discovery and development. When we talk about drug discovery and we talk about identifying compounds or pharmaceutical agents, not just things that bind well, we have these pharmacologists and medicinal chemists who play out in this area. So what they do is they look at how a drug binds receptor, how it can mediate some sort of biological mechanism or transduce some sort of an effect. And generally, after we identify some target, we do these hit to leads to try to identify if something is going to be valuable. We then go back and characterize the pharmacokinetics, and we try to develop a PK/PD type model so that we can explain and predict the effects. Well, as someone in drug delivery, when I worked in industry, we weren't really told to do that. We were told to get the most amount of drug in a formulation in the most stable form as possible. And I don't ever recall in any of our meetings being told to optimize exposure response. It was always, what's the most amount of drug you can actually have? So going through my graduate program, I was doing a lot of lipid-based or liposome drug delivery. We could see extreme differences from administering free drug versus doing liposomal drug, profound differences in the pharmacokinetic and exposure. But we never actually asked how we're controlling the rate of release or asked why we're actually controlling the rate of release. So this is what my talk's going to focus on. So the other thing I want to bring up is this shift from doing conventional medicine type approach to more of a precision-based medicine approach. And again, I work in the area of cancer, so the first thing I'll tell you is for most disease states, this is probably not the optimal thing. We always, in drug delivery and pharmaceutics, we tell people we have the best delivery system in the world. It's called a tablet. And we can deliver it for most agents, and for most disease states, the tablet works wonderful. But when you start talking about neurological diseases, infectious diseases, and cancer, the tablet doesn't work as well given some of the targeting and off-target toxicities. So when we talk about precision medicine, what we're talking about is instead of treating large groups and hitting everybody, what we're talking about is trying to identify specific people within the group so that we can actually design medicines more rationally that are going to target specific patients. And again, I'll get ahead of myself and tell you the FDA has recently approved drugs for cancer, and it does not say for breast cancer or for prostate cancer. It says for this specific molecular signature at this point. So rather than actually trying to classify that all breast cancers are the same or all prostate cancers, we're now looking at specific molecular signatures that we're going to target. So the one challenge, we use what it's called a multi-omics approach. And when you ask people what this actually means, when we talk about omics, again, the central dogma of biology is we start with DNA. From DNA, we basically transcribe it to RNA, then we translate it to proteins, the functional unit. The challenge with protein, though, is, again, not only do you have to make the protein, you have to traffic it to the right site within the cell. Sometimes they have to be post-translationally modified. So at any one of those steps along this process, you can actually have errors or defects, which then limit a particular physiological process challenged. So a lot of what we've been doing is doing genomics, where we actually look at the DNA. The bulk of my lab has now been doing transcriptomics, so we're actually looking at the RNA that's produced to look at how much is being transcribed. We are doing very little at this point. We're doing targeted proteomics. We're not doing widespread proteomics, although the capabilities exist. And then we're also doing some metabolomics. And again, my lab tends to be related to lipids, so it is more lipidomics, being more specific in that area. But just to give you an idea, when you start looking at all these omic technologies, the one thing that you have to do is collaborate, because not only as someone in drug delivery, I'm not trained in omics, but the sheer volume of data we're generating is incredible. So to give you an idea with transcriptomics, we were at a recent meeting, and we probably have about 800 different transcriptomic profiles now based on the different dosing schedules we've attempted. And again, when I go through my talk, you'll get a brief idea of some of the things that we're doing. And this is where machine learning and AI are going to become important, because as robust as my postdoc is, she still can't go through all the data systematically to look for some of these relationships. So I'm going to start, my lab is primarily interested in prostate cancer and breast cancer. This is one of the fun things when you get out of your postdoc, you try to set yourself up to be successful. I picked prostate cancer as a slow-growing cancer, and I picked breast cancer as a fast-growing cancer, until I started writing grants and realized nobody cares about slow-growing prostate cancer. They want actually fast, aggressive prostate cancers. So I have two cancers I work with now that are both fast-growing and both have different levels of hormone sensitivity related to them. But prostate cancer is the most common solid organ malignancy in men. It is the second leading cause of death in men. There is a number of risk factors, both age, race, ethnicity, and genetic factors. And with low-grade disease, patients typically live past 15 years, and they tend to pass away from some other disease state or comorbidity. They're not actually dying of prostate cancer. The treatment for this starts with active surveillance. They can then do a radical prostatectomy. There's radiation. We have both first and second line androgen deprivation therapy. The first one tries to knock out the androgen. The second line therapy actually targets the androgen receptor. The sad part is, for certain people of genetic background, they develop aggressive prostate cancer earlier in age. And then there are some risks. So African Americans tend to develop it at an earlier age, and it tends to progress much more rapidly. So my research is focused on aggressive, lethal, these castration-resistant metastatic prostate cancers. They tend to fail all therapy over time. They become resistant to the taxane, docetaxel, and cabazitaxel. For second-line therapy, the AR-positive cells, we have found that even though they block the androgen receptor, the signaling axis is maintained, which is still under investigation. Immunotherapy has been found to have some benefit, but only about 4 to 5 months. And the biggest challenge is, if you look at de novo, it's only about 1%. But after we treat patients with these second-line hormone blockers, what we see is the development of neoendocrine variant prostate cancer, extremely aggressive, rapid, highly metastatic. There's no current clinical treatment options for this. So what are some of the obstacles we have for treating not just prostate cancer, but cancers in general? There's a lot of heterogeneity, and I argue with people all the time, there is no such thing as a single breast cancer. Every woman who has breast cancer has a unique individual breast cancer. A lot like men who develop prostate cancer, they're all slightly unique, not only based on their genetics, but based on their lifestyle, the disease, what they've been exposed to. There is variable treatment response, and again, this can be everything from differences in metabolism to also differences in things they've been exposed to over their life and changes in expression of efflux transporters, normal metabolic metabolizing enzymes. One of the biggest challenges is development of drug resistance. So again, the docetaxel, cabazitaxel, and the second line androgen receptor blockers. So even though we can use these therapies and block those cascades, the cascades still tend to be active even though we can show we block them, which leads us to other questions about how is the axis being controlled. When I was in graduate school, you could get a paper if you did one PCR and did one gene. Now if you try to submit that, you're not going to get published. They want to see whole pathways or networks of genes and transcripts and how they're all being interconnected. We also do some work related to disparity, so we do look at African American patients, but some of the challenges is the data sets are actually really poor. So we use something called the Genome Atlas in the US. There's 840 something prostate cancer patients within that, but only 11 are African American, and they all have very early disease, so it's not really a good representation of their disease, nor the genetics associated with it. And I think one of the biggest challenges we see with most cancers is this idea of stemness, or this EMT, epithelial to mesenchymal transition within cancers, where the cells ended up reverting back to a more earlier stage and they become highly aggressive. They can invade and metastasize into other tissues. So one of the arguments we've been making is there needs to be new therapies to approach this. As I said, I'm a drug delivery person, and I was in school in the 90s when the EPR effect was first being published and described. The EPR effect is the enhanced permeability and retention effect. So at this time, there were people developing micro and nanoparticles, and everyone thought this was going to save the world, and we were going to develop new advanced therapies. And what it basically shows is that as the tumor is growing and undergoing angiogenesis, you form incomplete vasculature, chaotic vasculature, so you have all these defects that would potentially allow drug particles to accumulate perivascularly, and you could basically deliver drugs in a more targeted fashion. Clinically right now, for those who follow the field, there's a lot of debate about this because they haven't seen the clinical benefit that they expected. I don't really think it's because of the EPR effect. I think one of the challenges is a lot of the nanoparticles out there haven't been optimized appropriately, so they're really not getting the long circulation half-lives. They're not actually doing a lot of the benefits we see in the animal model. So this is one of the challenges, translating from animal to human. So this, again, has led to an explosion of nanoparticles and the development of a variety of drug delivery devices. Christy Ansel from UNC has been putting this together. If you look her up, if you look her up, she constantly is updating this on her website. But what's interesting is not only does she put when it was first described, but she actually puts FDA approval. And what you'll see is there's a lot more that were described and less that were actually approved. And I think this gets to one of the challenges we have in drug delivery. There's a lot of creative and novel delivery devices, but people aren't really testing them in the appropriate disease model or looking at using them for the right actual disease state. So again, they can use it for cancer, but what type of cancer, what stage of the cancer, and what are the genetics behind the cancer? So I'm going to focus on liposome-based delivery, and again, the focus of this talk is not going to be on the liposomes themselves, but trying to give you an idea of how we're looking to actually optimize these using an omics technology. So again, why do we want to do this? We want to be able to give more drug, and as we know, cancer drugs are highly toxic, so if you push the free drug concentration, you cause systemic toxicities. Back 30, 40 years ago, there really wasn't a concern with toxicity. The mantra was, patient's going to die, so it's okay to cause toxicity. Now, if you go to the FDA, for everyone who's doing regulatory affairs, first thing they ask you is, do you get QT prolongation? Are you actually causing a cardio effect? 15 years ago, they didn't care about that. It was, we're trying to look at drugs that kill cancer. If it damages the heart, it wasn't a big concern. We've had a shift in mentality now about not only does it have to be really efficacious, but we don't want to cause a lot of that off-target toxicity. So if we can turn around and encapsulate larger doses of drug and protect it from the rest of the body, this is one of the advantages of using particulate-based type drug delivery systems. I'm a strong advocate for using long circulating formulations. So again, this red, if you look down here, this is time in hours. This is blood concentration or blood levels. The red, if you give free doxorubicin, is rapidly eliminated. So from the bar, if you look 1 week after injection, we still have higher drug than when you first administered the free. The challenge is the drug is entrapped within the nanoparticle. So again, for those who do pharmaceutics, don't hold this against me, it's not bioavailable even though we gave it by IV injection. It's trapped within the particle. But again, the fact that they are circulating so long, we can actually, this is a brain tumor model that we used to use, we can actually see a 30% increase in survival. This is actually extremely significant because the free drug, the animals actually die 1 day sooner than if you do untreated in that actual animal model. It's a glioblastoma model. And again, here's just showing you, if you look at the brain, the blue area is because of the EPR effect. We administer Evans blue to the animals. It binds to albumin. Albumin doesn't cross the blood-brain barrier. So in this model where you see blue is where you have a compromise in the vascular barrier, which is associated with the tumor. So what are some of the advantages of the drug carrier load? We can protect the body. We can take advantage of this EPR effect. We can alter drug exposure profiles. And to me, I think this is what caught me the most when I went from an industry transition to an academic, is why were we not really trying to optimize exposure rather than just put the most amount of drug in? Because we can really, my collaborators in engineering have all these cool tools for altering release rates. But again, our focus had always been, how do we just stabilize the particles to get the most amount of drug rather than asking what's the right exposure profile? And then again, the last one comes down to enhance efficacy and reduce toxicity. So at the same time when I was finishing up my graduate work, some papers came out about anti-angiogenic therapy and metronomic therapy. And it was something I was very interested in because, again, when we talk about conventional therapy, we usually give bursts of high-dose therapy, and then due to toxicity, we have to have a drug holiday. When the patient recovers, we hit them with another sequence of dosing. And from that point forward, we're trying to basically cause cell kill. This typically works at maximum tolerated dose. We are typically working on highly fast dividing cells, and generally you see a lot of toxicity associated with this therapy. Well, the concept between metronomic or fractionated was we could give a lot lower doses. This is not traditional homeopathy where people say you dilute things until very low. This is actually giving measurable concentrations, but over a much lower concentration over a prolonged period. And what for me, being a drug delivery and training in kinetics, to me what this looked like is just giving a patient a very slow dose infusion rather than actually giving them a maximum tolerated dose. And what was interesting is it showed that you could basically deliver below the maximum tolerated dose. You didn't see that you delayed the emergence and development of resistance. We were able to see low blood levels without causing significant toxicity. We could target tumor cells, immune cells, and endothelial cells, so we were hitting more targets than just the cancer cell directly. So, and again, this is just trying to show you, if you look at the doxliposome, our formulation, we can end up doing a single dose and we have coverage over at least a month time where we actually have above these low micromolar concentrations. What you don't see is this is total drug. If we actually take out and ask you what's the free fraction of drug that's leaking out the particle, it's only about 1%. So again, while this here is very high, far below it is a much lower free fraction, metronomic or fractionated type manner. So I shifted and I did something, and again, this is one of those things that you do and then you look back and wonder why you did it. Topotecan was my negative control. It wasn't supposed to have this type of activity. and we weren't familiar with some of the literature at it, but it is a clinically approved drug for the FDA. It has been around a long period of time. The kinetics of it have been well established. It's potent. It has broad anti-tumor activity. It's a topo-1 inhibitor. Its mechanism of action is it intercalates between DNA base pairs with the cleavage complex. It can basically trap the topoisomerase, and it leads to cell death. Its primary dose-limiting toxicity is leukopenia, but some studies have shown that it actually has anti-angiogenic activity when dosed low dose or fractionated. So after we started doing our studies, we came across a number of these. So there has been clinical studies looking metronomically at topotecan given orally, and they have not done well. So they've had a couple responders, some non-responders, and then some non-responders, and then some people who the disease actually accelerated in aggressiveness. So it's one of the challenges when we submit grants. This commonly gets put in our face that, well, this is wonderful, but people have already tried this. Why are you pursuing this area of research? So our hypothesis was that we can use drug delivery approaches to actually control or regulate gene expression, both genetic and epigenetic influences, to treat actual castrate-resistant cancer by preventing both drug resistance and EMT progression. A number of objectives we had. We wanted to identify molecular mechanisms using a multi-omic strategy to identify specific disease characteristics, as well as what we don't normally talk about is what the drug carriers actually do to the disease in the absence of drug, which people don't realize. We're using drug formulations that are relatively inert. They are composed mostly of components that are found in your body, but when we form them as a nanocarrier, they do actually alter gene expression within our models. We'd like to utilize or identify and utilize molecular signatures to predict patient response. So, and again, for some of this is currently being done. We look at someone's molecular signature and we should not respond to a drug. What you don't see people doing is there a signature associated with how you give the medication versus should you give the medication? Again, what we're hoping to do is identify strategies that can lead to rational concomitant dosing schedules. And our overall goal was to achieve an optimal exposure within patients. So this is just to try to give you a little bit of a basic understanding of some of the complexity of prostate cancer. The initial phases, as I said, is under watch and wait. So this is the less malignant type. As we progress down here, you can put people on androgen deprivation therapy. They see response to it, and the disease comes down, but then they begin to develop resistance. From this point, you can give second-generation blockers. If you begin to show metastatic spread, we'll also put you on conventional chemotherapy, which again, the patients will then typically develop drug resistance to. All of the little bars here are just showing you different cell types. So the nice thing is we do have a diversity of cell types we can do experiments with that are hormone sensitive and insensitive. And then we have some of these fun ones here where our most aggressive disease, they're supposedly AR negative, but the one model that's clinically used as AR negative, when we test it, sometimes it's AR negative and sometimes it's actually AR positive. And either way, if we block it, we can still see some of the axis still signaling. So for someone who does a lot of in vitro studies, one of our biggest challenges was how do we look at low dose exposure? So you heard earlier today, they said trying to get around animal studies. The US legislator has recently put out a new law that says the FDA cannot mandate animal studies. And if you talk to people in Cedar and Sieber who do the drug development and biological development, they said it is all marketing and publicity because at this point you can't do these studies or generate the data we need to evaluate without doing in vivo studies. With that being said, I collaborate with Dr. Lipke, and some of the things they're doing in microfluidics and tissue engineering I think is going to help us identify some things, but not everything. But one of the challenges we wanted to do looking at conventional and metronomic dosing is standard assay, we seed cells 24 hours before we need them. They basically get to a certain level of confluency, and then 24, 48, and 72 hours, we do different things to them. So for a conventional dosing, you put the drug in at time 0, and then you read one plate at 24, another plate at 48, and another plate at 72. For metronomic, we would read one plate, at 24, but then another plate we would actually change out the media with fresh drug on it to simulate a certain MTT, SRB, MRS type assays that look at mitochondrial or total protein. And across all the cell lines, we showed even under a short 72-hour window, metronomically they tended to respond as well or slightly better than doing conventional therapy. We also did markers for apoptosis, and then we did a scratch assay with the cell migration. And again, the cell migration one again showed that we were able to slow the migration down using the metronomic relative to control and conventional therapy. To get back to drug delivery, why did this all start? Before I did all of this work, I had a very enthusiastic graduate student who came up, and at this point people are trying to make topotecan liposomes, and the problem is the topotecan, being a lactone, rapidly would pee out of the liposomes and leak, and it wasn't stabilizing. So what we wondered is, could we just do something a little backwards, and topotecan reversibly forms a carboxylate ion, and the carboxylate ion being charged doesn't cross membranes as well. So what we were able to do is force the drug into the lactone, load it within our particle, and then once it was in the particle, we could basically change the conditions inside the particle that had formed the the carboxylate to the inactive form, and it kind of got trapped. It's a lot like Doxil. People don't understand the reason Doxil is so stable isn't because we've remote loaded it. Liability within the particle, it precipitates out, so now it has to undergo dissolution before it can leak out of the particle. So our goal here was if we can trap enough of the carboxylate ion, when it leaks out, if it hits some of the a slightly acidic pH, all we need is a couple percentages to convert back to actually be therapeutically active. And the best part is anywhere the particle accumulated in a normal tissue, it would be relatively inactive because it was inactive drug. So as a way to reduce toxicity in addition to reverse targeting. The challenge though is we got to this step and he started running cell culture studies and everything looked great. And the question he asked me is, well, what's the release profile I want? Because he generated one profile, it looked great. Then he changed the cholesterol level. We played with the lipid compositions. We got another change in the profile of release. Activity, believe it or not, wasn't that different, but it was slightly different. And after he did like 5 formulations, he's like, well, which one are we going to go into the animal studies for? And at this point, we started to realize, we're really not optimizing or doing targeted type delivery. We're just guessing at what's actually happening. So to give you an idea, we decided we wanted to start experimenting to get a better understanding. We did 2 different things. One, we did direct tumor injections. One, if you're ever in a cancer center, the clinicians there do not like doing pharmacokinetic studies, so they do direct tumor injections. So if you're trying to publish in clinical cancer research or cancer research, it's acceptable to do that. As somebody in a college of pharmacy, we should all cringe with this because directly injecting into a sub-Q tumor is not the same thing as distributing through the body and reaching the tumor. So we did that, plus we also did injection, but I wanted to simulate long exposures. And again, I think we all know if you put a nanoparticle or any sort of particulate-based delivery, controlling it is very difficult. So we chose to use an osmotic pump that we could subcutaneously implant. My student somehow took literature values and simulated what drug concentration we wanted. And this is actually over 30 days. We actually wanted the levels to be slightly higher, but again, just from literature values and making some assumptions about absorption from the sub-Q, he was able to nail that we were somewhere in this range of being 10 nanomolar, which is what our 72-hour in vitro studies had predicted. So when we did these, we looked at control versus conventional dosing. And again, the little dots are when we conventionally dosed, the mini dots, this was the intratumor injection showing you the metronomic. And then down here, the bar is the continuous Alzit pump. And again, conventional dosing with IV. And again, this was done intravenously at this point. In all instances, we actually saw enhanced activity. What really was interesting, and this is where I tell my graduate students, you have to check each other's math. When people ask what's the difference between GLP and normal lab, when you do your calculation, someone checks it. Well, my one student prepared a ton of Alzit pumps, and we ran the first study in red. The next student, because I liked to add variability to the study, I had another student prep him. We did not check his math. he ended up preparing a more than 10-fold less. It's about 25-fold less. We didn't pick up on it. Well, the interesting thing, again, if you look at the blue is as efficacious, if not more. I mean, it's not statistical, but the animals lost even less weight and looked healthier than our typical metronomic dosing. And again, we based it on what we saw in vitro, which only went out 72 hours. This was actually below our limit of quantitation on our mass spec assay, so we couldn't even quantify it in vivo, but we did have all his math and we were sure of how he actually prepared the formulations. What's interesting though is when we did the histopath, we saw no evidence of actually inhibiting tumor vascular staining. We did not change angiogenesis, which is what we thought we were going to see. Our in vitro studies had showed we were changing angiogenic markers, but in vivo, we did not see any differences in actual blood vessel staining. So this is where our lab shifted over and started doing a lot more molecular work, and we started running these targeted arrays. And this is back before people were doing RNA-seq. So you could only get about 90, I think it was 94 genes, and then there was some control in there. And when we did this, we were able to actually identify a variety of genes. And again, I tell anyone who gets into this area, you're always going to find something. And the hard part is actually scanning the literature to determine how much sense it makes. Because again, there's certain things you should get based on the literature and what's known about the disease, and what you're interested in are the things that maybe you're not as familiar with, or things that might be slightly different. But from this, we were able to look at our dosing and compare two different types of cells and look at genes that overlapped. And again, then we were able to repeat that versus conventional and metronomic between different cell lines. And as I said, my students became a little aggressive and started running every single combination they could, to the point where we identified different signatures depending on the different cell lines we actually had. From that, again, we did a quasi-validation. So for the pathologist in the room, I apologize, this is not a medical validation. This was we saw genes go up or down, and then we did Western blotting or immunoblotting to look at expression, so it's validating what we saw in the transcriptomics. The interesting thing is, for many of the genes, if we saw the gene go down, protein level went down. We also had identified a number of them where they did not agree, and we saw differences where the transcript was greatly reduced, but the actual protein expression was significantly elevated in these cell lines. I think the key marker here, though, is what we found is that genes associated with EMT, drug resistance, cell proliferation, invasiveness were all altered relative to the metronomic therapy, so they were reduced significantly compared to conventional dosing and control. We use something called ingenuity pathway analysis, so everyone's talking about AI and machine learning now, but we've all been using it for years. If you've used a Microsoft product in the last 10 years, you were already using it. Their spell check was sort of using this. But the pathway analysis, if you put in a typical conventional drug exposure using topotecan, it will identify genes that are altered, and it will identify what disease states it should impact. You will never see prostate cancer identified. When we loaded the signatures, the profiles coming from our metronomic dosing, one of the top ones that got hit was prostate cancer. So even though traditionally pharmacologists have been using these tools for years, they give a dose and they actually look and see what the targets are. We're now giving a drug, but we're giving it at a very different exposure profile than is typically used, and we're seeing that other now targets are emerging. So, and again, when we did the pathway analysis, it said that the metronomic altered genes with drug sensitivity, angiogenesis again popped up, EMT, and it improved survival time within these cells. The one thing I'll point out, though, is we keep seeing angiogenesis, although we didn't see tumor vascular density staining. What we did realize, though, is there's lots of... It's interesting when we took our profile of genes that we said was important, and then we scanned it, did a cluster analysis in the genome, and what we found is, again, what we're saying is when we treated with metronomic, we altered the genes. Well, if you looked at patients in the TGA, so they've not had this metronomic, the ones that have our altered gene expression live significantly greater than the ones that have the control. So if you can, in theory, treat metronomically and shift their profile, in theory, you could actually see a significant enhancement in survival. And again, all we're doing is changing how the drug is being administered schedulally. It's not actually, we haven't done anything modifying the drug at this point. I mentioned we saw some genes that actually were significantly decreased, but the protein went up. So we ran microRNA arrays and did epigenetics. You don't see a lot of the epigenetics, the HDAC and the methylation, because none of that correlated. What we did find, though, is our microRNAs were significantly, or at least a few of them were significantly correlated with the genes and the proteins we saw differences in. And they're actually have been shown to be important in prostate and a variety of other cancers. So that actually was nice, because when you do these pathway analysis, it will actually identify things that don't make sense. And the first time we looked at it, a lot of ours did not make sense. And the reason why is that they're not used to being dosed that way. So in their data bank, they don't have any history of having these things changing. So we did a pan-cancer analysis, and again, it's not to have you look at all of the individuals, but each one of those is an individual cancer. And again, good and bad, we found that our profile correlated with aggressiveness in a number of cancers. It was 5 or 6, and it didn't correlate with other ones. And again, my student got all upset and said, oh, this is not good. And I said, no, this is good, because if it correlated with all cancers, we're probably chasing the wrong thing, because I don't believe in the magic bullet where one drug or one pathway is going to treat all cancers. They're different. So we have been able to start doing this and actually pull cells from other cancer lines and actually test them to look at them. This is where I think it gets more interesting, because when we first did this, my student designed some animal studies, and it wasn't too bad. He thought if I could order him about 6,000 mice, he could test about 5 formulations. And I said, we're not ordering 6,000 nude mice. NIH will not pay for it, and my associate dean of research is not going to give me the money to run a couple $1 million in animal studies just to test things. So what we did is we shifted to using a more advanced 3D modeling system, and Dr. Lipke is an expert in this, not myself. But what we found is we can actually grow on a more basic side. Our lab is doing these 3D spheroids, and we can grow them where now, instead of going out only 72 hours, we can do exposures for much longer periods of time. The other thing is we can also mix them, so this is looking at drug, but we can also mix them with fibroblasts and drug and turn around and see distribution of how things distribute through the spheroid, as well as the where they migrate through over time as well. This is something that, again, Dr. Lipke's group has used, and I've collaborated with her. It's something we hope to get into. It's using these in vivo or these physiological type chips, where they've actually sliced tumors. You can put a tumor in one situation, in one compartment, and then it can invade and migrate into other compartments. And you can see this for those that are engineering based. It'll actually show you the different flow dynamics based on the size of the vessels. Here again, it's better simulating what's in reality versus our standard plastic well dishes that we use. So for the students in the audience, I will tell you, I did not tell my students to do this initially. And you'll get an understanding why, because when I show this to people, people think I'm a very mean person. But I had one of these PharmD PhD students. He had just completed his PharmD. He was working on his PhD, had a much greater clinical understanding than I did. Well, he was interested in how the cycling could actually lead to resistance. So we took our spheroids, and again, we ran RNA-seq and a variety of other omic techniques on them, and we had baselines. We then formed 3D, we took the 3D, so we had the baseline on the 2D growth of these, and we had the baseline on the 3D growth of them as well. Not just after you form them, but form them, then analyze the RNA-seq after 1 day, 2 day, 3 day, 5 day, 1 week, 2 week, and go out. Well, what he did is he turned around and made 3D spheroids, and from the 3D spheroids, we waited a certain amount of time, and we ran the potency, doing a classic potency where we put drugs in, and then we do either an Alamar Blue or a standard type of cytotoxicity. What he also did is he took some and he let them just grow control. We took some and he basically did the conventional dosing, and then he did a metronomic fractionating dosing for 1 week. Now, most of us, we kill cells and then we look at them, and he didn't like that idea. So what he said is at the end of the week, to give you an idea, he had multiple plates for all these. He would take one set of plates and run RNA-seq on them. He would take another set of plates and take the 3D spheroid and run the IC50 on them. He then took another set of plates and took all the 3D spheroids and said, let's make them healthy again, dissociated them and grew them in 2D so that the dead cells would leave and the healthy cells would remain. He then would split those and do, again, IC50, and then he'd remake another spheroid and repeat this. So when this says 1 week, at this stage here, it took another 2 weeks before we get to here. So we originally discussed him doing this cycle for 2 weeks. Well, Josh was presenting and the data looked really good, and I wanted to say, you know, you should go out. And as a good graduate student, he said, well, I haven't really told you everything. I'm currently at 5 weeks cycling. So he'd been doing this almost 3.5 months, cycling these studies. We were able to turn around and compare the gene expression, and I'm not going to go through all these, but again, we found the longer we did the metronomic treatment relative to conventional, we saw a decrease in drug resistance. In the conventional ones, we saw a significant increase in drug resistance, significant increase in EMT and aggressiveness of the cell. And when you looked at here the sensitivity, conventional dosing by 5 and 6 weeks, these were 200 to 300 fold, or is it 57.9-fold higher in resistance relative to the metronomic dosing. What was interesting is for those of us who work in cancer, one of the ways to develop cancer cells or resistant cells is to low dose expose them, but that low dose is actually much higher than what we're talking about here when we talk about metronomic dosing. Just to be clear, we did end up working, because again, I'm not advocating we're just going to do metronomic dosing. We then took cells and treated them with docetaxel, which is the standard of therapy, and we were able to say with metronomic in this, we were able to improve its standard IC50. We ended up doing it in different cell lines. We have both an aggressive and a highly metastatic version. We also were able to turn around and look at apoptotic markers. We did flow cytometry, again, looking for EMT, and then we did a colony forming, and all of the data suggested that we could take these cells and basically treat them. We went the next step further, and we ended up doing the full RNA-seq on these with different exposure profiles. We saw a synergism related to this cell kill. The other thing that was interesting is we saw a whole bunch of efflux proteins that were altered with conventional dosing, whereas with metronomic dosing, it didn't seem to upregulate them. So we believe this is one of the pathways that's assisting for improving the therapy. Collaborating with another faculty in engineering, he makes microfluidics devices that have different size pathways, and then he can put the cells in and looks at how fast they migrate. So we were able to, and I'm not sure this will work, oh, there we go. So the one on the right is the highly metastatic, the 3M line, and it basically moves through those chambers much, much faster than the PC3. Well, we were able to turn around and do the metronomic cycling with these and then put them in, and we were able to show that we could really decrease the rate of migration. So not only were we not causing resistance, we were preventing them from actually being as metastatic or invasive as other cells. We ended up running, again, more RNA-seq. We were looking at different EMT markers. We were able to identify a variety of subtypes, even within certain cell lines, to show that there's differences. And again, where I think this becomes important is when we look at how therapy fails if we don't kill all of the cells, they can actually regrow into more aggressive metastatic. Our argument now is rather than try to kill them all, we should be doing a metronomic approach, almost, and I hate to say it this way, but like HIV, where they treat patients for very prolonged periods to keep the viral load down. In theory, we could do metronomic in combination with conventional therapy to knock them down. We then took our profile again, which every time we do this, we revise it, and we were able to show again with the markers we identified using individual ones instead of a large signature. We were able to show that they were clinically relevant. Well, again, this is not a clinical study, so if you're a pathologist, you're going to cringe when I say that. But we've also now used CRISPR to knock out the top individual genes. And what's good and bad about this is we're seeing that if you knock out one gene from a drug discovery perspective, it's a lot easier to develop a drug to hit a single target than it is to try to convince a bunch of clinicians we're going to metronomically dose people, especially since the way you dose, the patient compliance will be more difficult. But again, we were able to use CRISPR, knock it out, and then turn around and show that our metronomic dosing significantly matched after we knocked out the one gene of interest. So again, I'll end here. Our metronomic increased anti-tumor activity. We proposed a multi-omic strategy for student had done. A lot more omics and do that in combination with the suiting studies we typically do. Again, as I started off, I don't do this work anymore. When I go into the labs, my students kind of take me by the hand and try to lead me out of the lab because they don't want me touching stuff anymore. But one of the tops of this was Tarashi Ghosh. She's completed her PhD in Minnesota, came and worked in my lab as a postdoc. Josh Davis, he was the other student. He's currently at Moffitt. She's currently at Harvard. These are current students who are working on this. Again, when I list the collaborators, this is just a partial group. When I first started, I used to have 3 to 4 people on our papers. Now our papers have 12 to 15 just because none of us have all the expertise in all these components.
Concluding the session, prostate cancer is the second leading cancer of non-cutaneous cancer-related death in the US. Xenographic tumors. The combination of docetaxel and topotecan is effective against treatment-resistant prostate cancer. Using bulk and single-cell RNA sequencing and target genes expression profiling, they identify key genes associated with prostate progressions in European, American and African American subtypes and taxane resistance low androgen receptor MCRPC. They identify novel genes and potential biomarkers such as serpines which regulate prostate cancer progressions and drug response. And their findings suggest that a multi-omic strategy can improve drug delivery schedules and identify new therapeutic targets. So thank you, sir. Now moving up towards the questionnaire sessions. So anyone have any questions regarding this topic can ask the question.
Thank you, sir, for your fantastic research work. I congratulate you and your team for this fantastic work, and definitely we have got the new insights for this. But my question is, when you decide the dosage, you said that we purposely give the dose much lesser than what is been suggested. So how do we actually calculate this dose, and how do you have compared it with that Aljet? Because even in Aljet, was it the same liposomal drug delivery, or is it what the concentration of the drug which is been given? So these 2 points actually was not clear during your discussion, so just I wanted to know about it.
So at this point, we don't have a definitive answer. So actually, I was at a meeting a couple weeks ago, and when someone saw the transcriptome data we had, they are actually a bioinformatics person who has a machine learning tool. He wants to take what we have now and try to run simulations. And when I say simulations, they said that they're using, which one is it? I think Big Blue 2, the supercomputer. And he said if they run 1 million simulations and then turn around and tweak them, they should be able to see differences in gene expression based on what we have to try to narrow it down. The problem is, as much data as we have, it's really with only 2 different dose levels or doses we ran plus conventional dosing. So I kept telling him we're assuming a lot of linearities if we try to do that. But again, I think the modeling and simulation and AI, what it's going to let us do is develop new hypotheses to go back and test. Where we think needs to happen is instead of Josh running a bunch of, I think it was a crazy number, I think he did something like 696 well plates. Instead of doing all that with spheroids, what he needs to be able to do is we need to load spheroids into a multi-well format and do continuous flow through them so we can do them in real time relative to brute force. At that point, I think we can then optimize it. The formulation we had, the liposome formulation, actually leaked slightly faster than what we showed for the second Alsip pump. So what we need to do is actually slow our leakage rate down even further, and that's some of the next studies we're trying to do is get NIH to give us money to do the animals, because the hard part, which you didn't see, is all the controls for doing here. When you start changing out media, it can influence cell growth. So we had conventionals that had media swaps similar to metronomic, similar to conventional. So I think we had 12 controls for every one set we ran, which it's just very time consuming. But yeah, we don't, at this point, I was at the APS meeting in, I think it was October, and 2 companies came up immediately and they're like, what algorithm are you using to do this? And this is where I'm beginning to collaborate with computational. I do PK modeling, but this volume of data is way outside my expertise. So we're needing bioinformatics.
Anyone? Okay, first and foremost, I am express my gratitude to distinguished guest speaker, Dr. Robert Arnolds, for giving valuable insight, experience, knowledge and information among us. And we are honored to have you, sir. Thank you so much. And lastly but not least, I am thankful to the Multi Pharmacy College and especially Nulvi sir for inviting me in today's international conference for chairing this session. Thank you so much, sir.
Thank you so much, sir, for your valuable words. I kindly request Dr. Robert Rusty Arnold to mark his esteemed presence by signing the board. Thank you, sir. I request Dr. Nidhi Chauhan to felicitate Dr. Robert Rusty Arnold with a memento as a token of gratitude and love. Thank you, ma'am. I request Dr. Anand Deshmukh to felicitate Dr. Nidhi Chauhan with a memento for chairing the second session of the 7th International Conference. Thank you, sir. I request Dr. Uttam More to felicitate Dr. M.N. Mulvi, sir, for chairing the first session of the 7th International Conference. Thank you, sir. We have our next plenary lecture on the topic Improved Delivery of Liposomal Drugs in Cancer Nanomedicine by Dr. Jay Chandra Ramapuram, Professor and Graduate Program Officer, Department of Drug Discovery and Development, Harrison College of Pharmacy, Urban University, USA. Dr. Vinit C. Jain, Principal, Bhagwan Mahavir College of Pharmacy, will be chairing this session. So let's welcome Dr. Jay Chandra Ramapuram to deliver the lecture.
So very good afternoon to all. So today we are having Dr. Jay Chandra Babu Ramapuram, who is currently Professor and Graduate Program Officer at the Department of Drug Discovery and Development, Harrison College of Pharmacy, Auburn University. He has over 25 years of academic and industry experience in formulation and drug delivery research. As a principal investigator, he assisted pharmaceutical company in formulating water-insoluble drug molecule belonging to different therapeutic category. For this, he utilized innovative drug delivery platform such as nanoparticles, nanovesicular system, cyclodextrin complexes, melt execution system, and amorphous solids for delivery across oral, ocular, and topical routes and cancer tumors. He published over 150 peer-reviewed manuscripts and book chapters and over 125 conference abstracts. He is serving as a co-editor in chief for current drug delivery, associate editors for cancer reports. He is a long-standing member of American Association of Pharmaceutical Scientists and has served on many of the conference committee. So I request all of you to give him a big round of applause and invite him to deliver his talk on improved delivery of liposomal drugs in cancer nanomedicine. The stage is yours, please.
Good afternoon, namaste everybody. I know it's late and you might be hungry for lunch, and I try to make it straightforward, simple, and as much as possible I try to save the time to finish early if possible. Okay, so first of all I want to thank the organizers to give this opportunity to come back again and give my second talk this year, and I'm very fortunate to be here and glad to be here to meet you all again. So this is my another topic. This is another part of research that we have been doing in Auburn University. Last time I talked about 3D printed dosage forms, and liposomal dosage formulation is another area of my research that some of my graduate students have generated some data that I would like to present today. Okay, so I want to introduce life cycle management of drugs as a strategy to improvise the drug delivery and through the formulation approach that is followed by the pharmaceutical industry. I just want to introduce the term life cycle management, what it is, and how it is applied in relation to liposomal drug formulations. So life cycle management by definition is the strategy to maximize the value of existing drugs by extending the product life
Actually, this is a vital business strategy that companies follow to improve the profit of the pharmaceutical companies work on making the profit, right? In addition to helping the patients to save their lives, they also have to survive in the market in terms of sustaining the revenues by selling the drugs or selling the formulations, right? When you look at I try to make it simple in terms of understanding. When you look at the new drug product life cycle, basically, when the drug is introduced in the market, first of all, there are several phases here. You got early phase, middle phase, and late phase of the drug existence in the market. Actually, there is early phase is a pre-market stage where the drug is developed, drug formulation is developed, and get it approved by FDA. Then the product goes into the market. That is where the companies will start seeing the profits. Until then, there is a negative profit, as shown here. In the discovery stage, there is a negative revenue. And once the product gets into the market, you see the revenue goes up for a given drug or given formulation. Okay? So during that time, the revenue peaks up in the middle phase of the life cycle, and companies try to sustain it or plateau it so that they keep seeing the maximum profit, right? The idea is to get this phase going on so that they see the maximum profit. So how do you achieve that? So generally for any drug product that we introduce in the market, people buy because of its benefit, and after certain stage, the product there will be better product coming into the market or better drugs coming into the market, so that the product loses its interest. For example, if you take an iPhone, for example, any cell phone, right? In the beginning, people buy crazy. Later on, people get used to it and get bored up, and they wanted to try something new. As a result, the revenue goes down for the company. I'm not talking just about the pharmaceuticals, but in general, any product that comes into the market, any consumer goods also. Initially, the enthusiasm is very high, and enthusiasm goes down. The companies, what they do is they try to sustain plateau the market so that they can enjoy the profits, right? iPhone, for example, they keep on introducing the new models, right? Why do they introduce new models? They want to introduce something different at a given point of time. Therefore, people will keep buying the same material on and on from the same company, so that they sustain themselves. Likewise, even in pharmaceutical industry, when you introduce a product to the market, they want to keep the profit. Okay, so there is a dotted line here to extend the life cycle, so that the companies wanted to enjoy the profit. So I'm talking this in relation to the formulation approach, using the liposomal formulations, for example. How we do that? So we generated some preclinical data in our laboratory. Just I want to give that as an example. How can you prolong or extend the life cycle using formulation approach? Okay. So when you look at here, new drug product life cycle, forget about all the consumer goods and iPhones and other things, but we are coming back to the pharmaceuticals here, but I just gave an example. So new drug product life cycle is shown in the yellow boxes here. That is the preclinical development and clinical development until the product is introduced into the market. That is the time there is an investment phase or a negative revenue phase at that time. So if you can shrink or shorten this time and introduce the product into the market, commercialize the product into the market, as shown in the purple boxes. After the commercialization, then we extend the time. Extend means you prolong the plateau phase, as shown in the previous picture, so that you see maximum profit. It's basically a form of presentation that I have shown, another way of presenting the data I have shown in the previous slide. So you extend it so that companies will reap the revenue benefit. So there are different type of approaches here. This is the time stamp indicating the number of FDA product approvals. As you can see here from the picture, there are different approaches like new formulation approach and new molecular entity introducing approach and new combination approach, new indication approach, and over-the-counter drug switch approach. For example, when the drug is introduced into the market, basically people keep buying, and later on they found the product, their different new product came or different alternative products came, and people enthusiasm decreases. At that time, companies, what they do, they introduce the new formulation. For example, ondansetron, oral tablet is available for vomiting prevention. The same company introduces oral film or orally disintegrating tablet. They're all the same drug in a different formulation. That's what is called as a new formulation. Companies introduce new formulation, or companies introduce new molecular entity belonging to the same therapeutic category. Our companies introduce new drug combination. Ondansetron in combination with something else. I do not have a good example. Then a new indication. For the same ondansetron, people are currently working on using it for Alzheimer's disease, for example. New indication for the same drug. That is the way they increase the life cycle of a given drug. And of course, what is the product use? Prescription drug in US, nothing can be sold without the prescription. Of course, same is true in India also, but it is strictly enforced in the US. So basically, then you introduce the same drug in the form of a OTC after establishing the safety, and people are not going to die by taking the medicine without a prescription. They introduce OTC medicines at the typically at a lower dose. So these are the different approaches. So we are going to discuss some data in relation to how liposomal formulation modifications can be used in terms of extending the life cycle of a liposomal drug. There are several different liposomal formulations which are available in the market, as shown in the picture. This table shows there are around 23 drugs which are available as liposomal drugs, out of which at least half of the drugs belong into category of cancer therapeutics. So there are several drugs which are liposomal. This is all published information which are available in the literature. And then there are additional the previous one, you got both the natural lipids as well as the semi-synthetic lipids here, and then you got additional lipids like such as DOTAP, DOTP-MA, and cholesterol and anionic lipids, cationic lipids as shown here in the picture. These are all used in the formulation of liposomes. Okay, so again, there are several drugs as shown in the list here, which are available, which are sold in the market as liposomal formulations. The reason I brought this table here into the picture is show the lipid composition. As you can see from the lipid composition, liposomes have got just the lipid, drug, and water. It's a very simple formulation. The lipid composition, as you can see, DSPC, disteryl phosphatidyl and cholesterol combination. There are only two lipids here. Liposomal formulation is very simple in terms of the composition. In terms of the formulation is complex, but the composition is very simple. It doesn't have too many ingredients like tablet, for example. So likewise, you see different commercial formulations or different drugs are made as liposomal formulation using different lipids. And composition again is very simple. You got either two lipids or at the maximum two or maybe three lipids, right? As you can see in the picture. So what we thought in our lab, can we structurally modify the liposome composition to come out with a new formulation with a better clinical efficacy? So that was the idea that we worked in our lab. So we modified the lipid composition because the composition is very simple. Can we introduce another lipid so that we can come out with a better formulation? At the same time, formulation is also safe. That was the idea. Okay, so we thought of working with ceramides. Why ceramides? Ceramides are biologically occurring lipids. Ceramides are within our body. Ceramides are everywhere in the body. Ceramides are which are essential for biological functions in the body. ceramides are available in the skin, ceramides are available in the brain, ceramides are available in the muscles, they are everywhere. So that means they are safe. Can we use ceramides to structurally modify the lipid composition to come out with the new formulations? So there are 9 different ceramides which are shown here, which are also listed as ceramide 2, ceramide 5, ceramide 3, ceramide 5, 1. You see different ceramides have different names. What does it mean? Like you see, ceramides are ester conjugates between a fatty acid and a sphingosine base. You got a fatty acid conjugated to sphingosine base. There are 3 different fatty acids here, like non-hydroxy fatty acid, hydroxy fatty acid, and then you got esterified hydroxy fatty acid. There are 3 different fatty acids, and they conjugate or esterify with a different type of sphingosine bases, such as sphingosine, phytosphingosine, and 6-hydroxy. These permutation combinations of 3 fatty acids and 3 sphingosine bases results in 9 different ceramides. So we have used in our research ceramide 6 and ceramide 8, which are short chain fatty acids, which are used in already existing liposomal formulations to structurally modify the composition of the liposome to come out with a new formulation which is safe, and wanted to see if it is going to be effective or not. In terms of the biological function, that I already told, the structural components of the cell membrane, that are essential for cell function. They serve as a carrier or transporter. They protect the cell surface by forming a stable lipid bilayer, and they modulate the process in terms of the cancer. They modulate the functions such as apoptosis, cell growth arrest, and inhibition of cell division, and so they serve both as a structural role to keep the structural integrity of the cell membrane, and also the functional role in terms of signaling. They serve as intracellular signaling role. So there is a rheostat shown here, rheostat of ceramide, and sphingosine 1-phosphate is abbreviated as S1P, right? So when there is a equilibrium between ceramide and sphingosine, and the equilibrium is shifted towards basically the ceramide and S1P ratio, when it is increased, there is apoptosis, cell growth arrest, and inhibition of cell division. And when it's a kind of a balance between these two, balance between ceramide and sphingosine 1-phosphate, and the balance is shifted in the ceramide to SNP1 ratio decreased, then there is cell proliferation and cell survival. This is kind of a constant equilibrium process that goes on in the body. The reason I brought that here into picture for the discussion is because ceramide, when it is present in the formulation, ceramide is known to cause apoptosis. Ceramide itself is biologically active in terms of preventing or arresting the cancer cell growth. So by increasing the equilibrium towards the positive, towards increasing the concentration of ceramide, we can achieve apoptosis, growth arrest, and inhibition of cell division, etc. for the cancer cells. So we have introduced ceramides into the liposomal formulations based on that reason. So how ceramides basically, they get integrated into the liposomal structures, and when they are integrated into liposomal structures, they are biological friendly because they are already part of the cell structure. So therefore they can better associate with the cell membrane, and there is a ceramide which is present in the liposome causes lipid rearrangement within the cell membrane. Therefore the drug permeates better across. Therefore there is due to lipid rearrangement, drug from the liposomes permeate better across the cell membrane due to presence of ceramides. Okay, so the difficulty with delivering cancer drugs is that the drug alone is very toxic to the normal cells. In cancer cells, cancer tumors, as well as the normal cells, the drug is very dangerous or very toxic to the normal cells in the body. You must basically make the drug to selectively deliver to the cancer cells. Therefore, companies came out with liposomal formulations. The drug is protected by liposome and delivered only directly to the tumor cells. Therefore, normal cells are not exposed. So the challenge is this drugs is a slow diffusion through the plasma membrane, and the drug itself is even if the drug gets permeated into the cancer cell, due to efflux transporters present in the body, basically the drug is thrown out of the cells due to efflux mechanism. However, the drug is present in the ceramides. Basically, ceramides, the drug is present in the liposomes. When the liposomes get inside the cells, the efflux, they are not available for efflux transportation to throw to eject outside the cells. Therefore, there is a benefit of formulating these drugs in the form of liposomes. Then we wanted to introduce ceramides to achieve better biological function. So, okay, we made the ceramide-based liposomes. Our idea is to deliver ceramide and anti-cancer drug in a liposomal formulation for better therapeutic effect that I have shown, I have told you earlier. So how we made the liposomal formulations? We followed the conventional method. This method is already being used by pharmaceutical companies. So simply we made the liposomal formulations by making this schematic shown here is called as thin film hydration technique. We take the ceramide, we take the lipids in a organic solvent, and then we do the flash evaporation. We make a thin film by removing the solvent. Then we hydrate it using the aqueous buffer, and when we hydrate it, we get the liposomes. So we get the coarse liposomes, because liposomal structures have got a size which is nano size. Basically, their size is around 200 to 300 nanometers. What is shown here got several micrometers. By basically take this lipid and extrude between two syringes in a lab scale. In a large scale, basically they follow different microfluidic technique to simulate or to achieve the liposomal formulation. But in the lab, we can follow the extrusion technique to get the liposomes in the nano size range, similar to what we do in the commercial large scale. So we followed this technique to make the doxorubicin and ceramide incorporated liposomes in the lab, and so here is the details of formulation that we have followed. We took a cationic lipid and cholesterol and another lipid called DSP lipid, and we made a thin film, or then hydrate it to get the coarse liposomes. Then coarse liposomes are extruded to get the fine liposomes or the nano liposomes. So the different formulations have been prepared and characterized. I'm not going to go into the details in terms of different formulation compositions. All I wanted to tell here is we altered the lipid composition, and we altered the cholesterol concentration, and we used the different cationic lipids here and different type of lipids that we have used to come out with different formulations. As shown in the picture here in the second column, all the formulations have got the particle diameter in the nano range, below 200 nanometer range, and the zeta potential indicate the stability of the liposomes. You need to have high zeta potential beyond the zero. I mean, zeta potential close to zero means there will be clumping occurs and the formulation get precipitated. You need to keep the zeta potential away from zero, either towards the positive side or negative side. Then the formulation is going to be stable. Therefore, we measure the zeta potential here. As you can see, zeta potential is much away from zero. That means the formulation is going to be physically stable and no clumping or precipitation occurs. And encapsulation efficiency, what is meaning by encapsulation efficiency? The drug is included. Say if you take, for example, 100 milligram of the drug, how much amount of the drug is loaded into the liposomes. That is calculated in terms of the percentage. So you see maximum amount of the drug is loaded inside the liposomes. That means there is less wastage of the free drug. All the drug is associated, whatever the drug that we have taken into the process, the drug has gone into the liposome structures. So we characterized all these things. Then how did you determine the free drug from the encapsulated drug? We followed the gel permeation chromatography, which occurred the principle that the drug which is encapsulated in the liposomes get eluted first, followed by the free drug, which takes longer time. It's like a molecular sieve mechanism. So the free drug get trapped inside the porous structure of the gel and leaving the liposome to come out first. Therefore, whatever the drug that you see here, so we have done different parameters for the liposomes that we found. We prepared the liposomes as a function of polyethylene glycol concentration, cholesterol concentration, and ceramide concentration. We use different ceramides. So this graph I am showing here, without boring you here, is to indicate that we wanted to eliminate free drug from the liposomal encapsulated drug, so that therefore we can calculate the encapsulation efficiency. Therefore we followed the GPC technique to get rid of the free drug, so that we know how much amount of the drug has been encapsulated in the liposomes. So the point I want to make it clear is that there is very less amount of the free drug in the liposomal. This is the control. When the drug is not included in the liposomes, this is the peak that you get. When the drug is included in the liposomes, all the drug get into the liposomes and there is very minimum or no amount of the free drug present. So that's the reason gel permeation chromatography has been used to know the presence of unencapsulated drug with the liposomes. So we did the in vitro release profiles for the liposomal doxorubicin formulations. As you can see, the drug, I don't want to again discuss the details in terms of formulation. I want to give you the big picture from the release studies. So as you can see, as a function of cholesterol composition, we needed at least 40% of the cholesterol present in the liposome to maintain the integrity of the liposomes. And we got 20 and 40%, and 40% the release is very sustained. When the drug is not present in the liposomes, all the drug is dumped within 2 hours. Liposomal formulations has prolonged the release up to 48 hours. Only about 60-70% released within 48 hours. The release is sustained due to presence of liposomal formulations. So the point I want to make here is that when we use the ceramide as additional ingredient in the formulation, we did not significantly alter the release profile of the commercial formulation. We took the composition, I remember I told you, we took the commercial liposomal formulation composition, then we added the ceramide. When we added the ceramide, it did not significantly alter the release profile of the commercial formulation. Then we did the cell viability assays. As you can see, ceramide-based formulations showed very highly increased cytotoxicity for the doxorubicin compared to drugs without the formulation without the ceramide. So this is the formulation, for example, this is the formulation without ceramide, and this is the formulation with ceramide. Say for example, especially C8 ceramide and C8 gluconic ceramide, both of them showed highly potent effects compared to the formulations without ceramide, which is also substantiated by fluorescence microscopy studies, where it is shown that DOTAP C8 ceramide formulation with integrating ceramide in the formulation showed significantly enhanced uptake by the cells, doxorubicin, compared to the DOTAP formulation without ceramide. Okay, so likewise we used we previously we only used we used skin cancer cells. We wanted to know, can it be the same process? Can we get the similar benefit when we change the cancer cell line? Then we have tested we have repeated the studies using prostate cancer cell lines. We got the similar effect. Again, ceramide loaded formulation has shown significantly enhanced cytotoxicity compared to the formulation without ceramide as compared to the controls. And fluorescent microscopy studies again demonstrated that the ceramide loaded liposomal formulation shown enhanced enhanced cell uptake of the doxorubicin compared to one without ceramide in the formulation. Likewise, we got similar results obtained with breast cancer cell lines. So we have done the studies with skin cancer cell and melanoma cell lines and prostate cancer cell lines, and finally we did the breast cancer cell lines. We got again similar effect as that of other two cell lines. The ceramide loaded drug formulation, liposomal formulation shown significantly enhanced cytotoxicity compared to the formulation with no ceramide. Okay, so as shown in the fluorescence images as well. Okay, so this is the summary of results of ceramide liposomal formulation studies. So doxorubicin liposome formulations exhibited significantly higher cytotoxicity effect compared to three different cancer cell lines. And in future we are going to do formulations correlated with doxorubicin and ceramide to see if it can produce significant benefit in terms of the cytotoxicity in the tumor-bearing mice. Okay, so another scenario here, and it takes another few more minutes to describe another formulation approach, how it is going to be beneficial in terms of enhancing the life cycle management strategies for the liposomal formulations. So ceramide is one approach. We can increase the potency of the liposomal formulation. Now we use the cyclodextrin formulation approach to increase the potency of the liposomal formulation, the second type. So I got short data to share to show how the cyclodextrin was beneficial for the liposomal formulations. So we have taken hispalon as an example. Hispalon is a polyphenolic compound with a potent anti-cancer activity. But however, the hispalon has got two different drawbacks. One is poor solubility, which prevents the loading of drug into the liposomes is difficult. Second one is hispalon is also a candidate for efflux transport. Therefore, the drug cannot penetrate to the cell that effectively. So therefore, by using cyclodextrin formulation, liposomal loaded hispalon in the presence of cyclodextrin, we can address the issue of the solubility for the hispalon. At the same time, we can also increase the potency of liposomal formulation due to presence of cyclodextrins. So, just ignore the data that present in the slide. What I wanted to tell from this slide in the big picture is that we already established hispalon is a very potent compound. When we used hispalon alone, without any formulation, just hispalon was tested in the melanoma cell lines, that we established the mechanism of hispalon effect on the melanoma cell lines to show that hispalon is very potent compound to kill the melanoma cell lines. So various mechanisms have been shown here, and hispalon acts by increasing the ROS production and increasing the nitrate content and increasing the lipid peroxidation and decreasing the complex 1 and complex 4 activities. So these results suggest that hispalon causes apoptosis, oxidative stress and mitochondrial function. So using this data, we thought that we can formulate hispalon in the form of a liposomal formulation. The neat hispalon is proven that it works very well. Next step is to make the formulation. So we made hispalon cyclodextrin based formulations because without cyclodextrin, if you make the Hisplan liposome formulation, the loading of drug is difficult. Therefore, we use the cyclodextrin to solubilize the liposome. The soluble complex has been included into the liposomal formulation. So Hisplan formulation approaches, we have taken directly, we formulated Hisplan into the liposomes, number one, but then we found that the drug loading is very poor when we did that. So then we thought in the second approach, we thought of doing first make the Hisplan cyclodextrin complex, then include it into the liposomal formulation. So we got better results with cyclodextrin based liposomal formulation for Hisplan. Okay, so this picture is very important, where I have shown that the yellow dots here show the Hisplan cyclodextrin complex. Hisplan cyclodextrin complex. If I did not make the Hisplan cyclodextrin complex, try to include into the liposome structure, what happens is the drug cannot be included into the lipid bilayers that easily. Actually, so when we make the liposomal, when we make the Hisplan complex, when we do not make the Hisplan complex, what happens is the Hisplan gets trapped into the bilayers, never get into the aqueous compartment. Whatever the drug present in the bilayers get leaked out very quickly. So therefore, it is important to make the Hisplan cyclodextrin complex so that the complex will lodge into the aqueous bilayer of the liposome, and the drug gets slowly released from the aqueous compartment through the lipid compartment into the cell environment. So therefore, we formulated cyclodextrin Hisplan complex first, then included into the liposomes at the second stage. So we hypothesize this approach improves the drug loading and release control compared to the liposomes made without the cyclodextrin complex, conventional liposomes. So in order to do that, we first characterize the complex formation by using the phase solubility studies to show that presence of cyclodextrin substantially increase the solubility of the Hisplan. We use two different cyclodextrins to show the increase in the solubility for the Hisplan, and there is a linear increase in the solubility of Hisplan presence of two cyclodextrins. Then we established through the J plot analysis that Hisplan forms 1:1 ratio complex with sulfur-butyl ether cyclodextrin, which is better, it produces better solubility compared to the hydroxypropyl beta cyclodextrin. Then we found that sulfur-butyl ether beta cyclodextrin has got the higher stability rate constant compared to the HP beta CD, and the complexation efficiency is much higher compared to the hydroxybutyl beta cyclodextrin. Therefore, we use SB beta CD in the formulation. Okay, then we did the liquid state characterization followed by the solid state characterization for the complex, so that the complex is fit for including into the liposomes. So solid state characterization indicates here that cyclodextrin forms, SB beta cyclodextrin forms a stable complex with Hisplan. So after proving that, then we made the formulation. So we know we have prepared this Hisplan cyclodextrin complex first, then we included that into the liposomes. So here is the two-stage process. First we made the Hisplan cyclodextrin complex as shown in the picture. We made the thin film hydration here. Once we made the thin film, then we didn't make the thin film here yet. For the drug cyclodextrin complex, we dissolve the drug and cyclodextrin in a solvent and lyophilize to get the complex. Then the complex is used in the liposomes, where we followed the thin film hydration method. We first made the thin film by using the lipid and solvent and evaporating the solvent and get the thin film. The thin lipid film is hydrated to get the liposome, coarse liposomes, which are subjected to extrusion to get the, not extrusion but sonication, to get the Hisplan cyclodextrin liposomes. Once we get the Hisplan liposome cyclodextrin complex liposome, then we did these are the compositions that we have prepared for the Hisplan liposomal formulations, where we basically prepared different type of formulations here. liposomes alone, conventional liposomes similar to the commercial formulation. Then we made the Hispulon loaded liposomes alone without the cyclodextrin. Then this is actually cyclodextrin liposomes serve as just control. Then we got Hispulon cyclodextrin liposome formulation and Hispulon complex liposome formulations. So these are the different formulations which are tested in the cytotoxicity studies, and the idea is I'm showing is here is that the formulations have shown as shown before the particles, all the liposomal formulations have the nanoparticle size range, and the zeta potential is away from zero, showing the stability of the liposomes. Then we tested the osmolality and encapsulation efficiencies, which is very much higher compared to the conventional formulation. Then we did the release studies for the liposomal formulations, and shown that the release is very controlled when we did liposomal formulation. We achieved the release profile sustained for a period of 24 hours, right? When we increase the Hispulon loaded cyclodextrin formulation to 20%, we obtained very sustained release formulation. So all these formulations, we did the cytotoxicity studies. In those studies, we have, as you can see, that Hispulon cyclodextrin complex has shown potent increase in the cytotoxicity for the melanoma cell lines. Very potent increase in the cytotoxicity, as you can see. I got the IC50 values, I will show in the next slide. And Hispulon cyclodextrin complex alone, the good news for us is Hispulon cyclodextrin complex alone showed significant increase in the cytotoxicity for the Hispulon. Then we included his HSC complex in the liposomal formulation in the right side, and as you can see, Hispulon loaded cyclodextrin formulations have shown very high increase in the cytotoxicity for the Hispulon. Here are the IC50 values. Okay, when we made the Hispulon solution alone, IC50 value is 14.1 micromolar. That means we need to use large amount of Hispulon to kill the cells. So by formulating Hispulon cyclodextrin complex, these are all compared at the equimolar concentration. Okay, Hispulon cyclodextrin complex have shown, see, at least 6 to 7-fold increase in the cytotoxicity for the Hispulon, which is a good news, right? We are able to decrease the concentration from 14 to 4 micromolar concentration. Then we made the liposomal formulations. As you can see, the cytotoxicity is increased by at least 2-fold for the liposomal formulations compared to the Hispulon solution alone. So in summary, we are able to make the Hispulon cyclodextrin complexation for the Hispulon, and included the Hispulon complex into the liposomes to increase the cytotoxicity of Hispulon by liposomal approach. That is where we can get the additional benefit of advantageously modify the composition of the liposome by using the cyclodextrin in the liposome formulation. And in the future, we plan to co-deliver Hispulon cyclodextrin complex based liposomes along with the doxorubicin, which is already available in the market, to achieve additional benefit of lowering the doxorubicin concentration or doxorubicin dose in treating melanoma cells or other cancer cells. So we need to, of course, perform in vivo studies to prove all the in vitro data that we have generated in the lab so far. So I want to acknowledge my graduate students who have generated this data. So I want to thank Ishwar who worked on Hispulon, and Manjusha, Noor, Chu and Chi. They all contributed the data which I presented today. I also want to thank my collaborators, Dr. Rusty Arnold, Dr. Amit Tiwari, and Dr. Padma Bhaskaran, and Dr. Candace Lyman from Vet School. And these are the past graduates and postdocs that who worked in the lab. I want to thank funding and other people who assisted this project. Thank you very much.
we are running short of time, so I will request the audience to have the interaction during the lunch session. So really, sir, it was a wonderful talk in a short period of time. You have really justified the topic on improved delivery of liposomal drug in cancer nanomedicine. So you have given the two examples that how this ceramide and the cyclodextrin complex, by using these, how we can improve the liposomal delivery. So here for the student, I will just say that this two-day international conference which is organized in the Dhanvantari campus is really, it will be a landmark for your future. I think you will be interacting with the 6 international experts who are there, who are experts in their research of liposomal drug delivery. So I think this can create a landmark for all of you. You can think of making your research further studies under their leadership. So I congratulate Dr. M.N. Nulvi sir for gathering such a good experts for this two-day conference, and I thank the
Dr. Jayachandra Ramapuram with a memento as a token of gratitude and love. Thank you, sir. I request Mrs. Sonam Gandhi to felicitate Dr. Vinit Jain with a memento for chairing the 3rd session of the 7th International Conference. Thank you. Now it's time for the lunch break. The session will commence after 30 minutes.